ठीक, हेलो। ठीक, ऑडियो ऑडियो चेक करो, ऑडियो चेक करो। ठीक, हेलो हेलो। ठीक ठीक ठीक, हेलो हेलो। ठीक, हेलो। लैपटॉप मेरे, ठीक है।
Wonderful deliberations from experts who've come from all over India to participate in this national conference. For this session, I would request Professor Sadiq Bopunia Patel and Dr. Mumtaz Ahmed and Dr. Anwar Ahmed, who are the chairpersons for the session, to please grace the dais so that we can begin. Yeah. Dr. Patel is here. Yeah, please, sir. Professor Patel is Professor Emeritus, former Vice Dean, JJ Hospital, former HOD, Department of Pharmacology, and also former Project Head for RRIU Mumbai. Dr. Mumtaz Ahmed is Deputy Director, Regional Research Institute of Unani Medicine, Patna. And Dr. Anwar Ahmed is the Assistant Director, Unani, from our National Institute for Research in Unani Medicine, Hyderabad. Moving with the proceedings for the day, we have lectures from MIT Institute of Noida, 2 of them from MIT Institute of Noida, 1 from National Research Institute of Unani Medicine, Hyderabad, and 1 from IIT Mumbai. We have Dr. Azizuddin Khan from IIT Mumbai. Now I request Dr. Nitin Sharma, who is the Head of Department, Professor, MIT Institute of Pharmacy, MIT University, Noida, to present his talk on colon targeting of Unani medicine via gum-based oral formulations for effective treatment of IBS, clinical intervention by gamma scintigraphy. Dr. Nitin. Okay. There is some technical issue. I think we can start with Dr. Navneet Sharma and then Dr. Nitin will follow. Dr. Navneet Sharma is Assistant Professor, MIT Institute of Pharmacy, MIT University, Noida. He will be delivering his talk on novel Unani medicine, infused organic oil-based insoles for diabetic foot care. For now, I think it's over to the chairs. We don't have a mic. Okay, I'll do so. We don't have a mic. I'll just call for a hand mic.
On this very good morning to all the
hand mic
from all over the part of the country. For this particular day, we celebrated the Unani Medicine Day yesterday, and today we are continuing with the research activities which are going throughout the country.
Excuse me.
I am very thankful to my 2 esteemed colleagues, Dr. Mumtaz Ahmed sir and Anwar Ahmed sir, for their presence here, and all the people who have come here, the research scientists. Definitely the number doesn't matter. It is how much the depth of the research that is there that is more important. Albert Einstein has said, someone has said, the great scientists are the great intellectuals. But he said, I don't agree with this. It is the integrity of the indigenous research scientist which makes a great scientist. So that is the integrity is most important. The number is not matters much. If one person is having integrity for his research, he will go to the level where it will be useful to all the mankind for years together. This particular Unani system of medicine is cultivated and it is more progressing in the country. It is because of the forefathers. I remember 2 names. One is the Hakim Ajmal Khan, who was the president of the Indian National Congress. Not only that, a freedom fighter, and he revived the Unani medicine in this country because the youngsters, they should know about it. And the second, the former director who established the CCRUL, the Hakim Abdul Razak. With 2 esteemed persons who knows better than me about him and all. So we start the session. And this is the first. I invite the first speaker to come and express his.
Good morning, respected chairperson, distinguished delegates, and colleagues. Today I will present our transitional research work on a novel therapeutic medical device and Unani medicine infused organic oil-based insole designed for diabetic foot care. So, diabetic foot ulcer is not merely a wound. It is a multifactorial pathology involving neuropathy, ischemia, infection, and abnormal pressure distribution. Despite available treatments, recurrence and non-healing rates remain high because current interventions which are available either focused on the wound management or improving the biomechanics. So, and there are very rare formulations which are available which both work on simultaneously on both the mechanisms. So our objective was therefore to integrate, to make a common platform which involves the traditional pharmacology knowledge, biomechanical offloading, and as well as sustained drug delivery. So together combines into a novel drug delivery system into a form of the insoles. And in the coming slides, I am going to explain like what all research aspect I involve in the development of these bioinorganic insoles. No, sir, that one is. There is some problem in projecting, no issues, ma'am. So just coming on the topic, I will just give you a glimpse of the topic. So as we know that human plantar surface is a unique therapeutic site. When we talk about the drug delivery system, no, sir, it is on the desktop. Anyhow, so coming on the topic. Yeah, this one. So second slide. Yeah, that one only, sir. That's fine. So as we know that the human plantar surface is a unique therapeutic site. It has continuous contact, controlled occlusion, and stable temperature, making it suitable for sustained transdermal delivery. So we develop a biodegradable insole that simultaneously redistributes plantar pressure, release medicinal oils, and prevents microbial colonization. So the innovation lies in combining two traditional independent domains. As you can see here, I took two traditional independent domains. The first one is the therapeutic insoles, which we use regularly. Previously also there are several therapeutic insoles available in the market, and those insoles are basically polyurethane based, and basically the sustainability is one of the aspect of those insoles. So what I have done, I had chosen the pre-existing moieties which are available there, and particularly the pre-existing oil. So in particular in this insoles, I use the mustard oil, and this is in something amazing, like I use 98% of the mustard oil in this. And there is no urethane compounds and the polymeric compounds are there, only the cross-linking agents and the mustard oil, that organic oil, which I use as a base in the terms of the insoles. And particularly, another aspect is the incorporation of the Unani topical formulation. So it is a combination of the insole, organic insoles, and as well as the Unani topical formulation. I incorporated that particular formulation inside these insoles. So conventionally, what we found, like topical medicines suffer from patient poor compliance. Poor compliance, that is the patient compliance, one of the important aspect when we talk about the drug delivery to the patients. Several times it was observed that patient used to forget to use those topical formulation. So by embedding medicine into a bearable material, therapy becomes passive and continuous. So this is the one of the aspect of this. Sir, please change. So when we talk about the transitional value, so diabetic foot management is lifelong, and preventive care is more important than episodic treatment. So episodic treatment is one of the important aspect, and currently what we observe in terms of the treatment of these kind of neuropathic or diabetic sores. So the common approaches or current approaches on which the most of the physicians rely is on the dressings, use of the antibiotics, and debridements. So all these, as we can observe, all these interventions are reactive in nature. So our device shifts management moreover towards the preventive palliative care. So we are focusing on the preventive palliative care rather than the conventional. Of course, we can use the conventional formulation which can use topically along with this preventive palliative care. Yes, shall I continue? Say fine. Ji, please. So, sir, coming on the next slide, please change. So coming on briefly I will explain about the strategy which I use. So here I divided my work into the two parts. The first part is the developmental part, another part is the transitional part. So in the developmental part, I use the computational aided drug design technique and as well as FDA approved response surface methodology to develop the prototype. Once we had developed the prototype, then we focused on the field-based usability trials, which includes the industrial scalability and technology transfer. So in this particular phase of our project, we are particularly focusing on the preclinical aspect as well as the development of the industrial prototype, as well as particularly focusing on the location ways, logistic ways, and the production ways. Like when we are going to the production in the industry, so whether it is easy to produce them within the purview of the economy or within the purview of their spendability. So this is the important aspect which we focused. Sir, please change. Please change. So coming on the structures, so the insole consists of cross-linked polymeric gel matrix, encapsulated therapeutic organic oils, and the most important what state of art is involved here, we use the encapsulation techniques. Rather than the verse release, we focused on the encapsulation technique driven by the pressure, which is impart on the sole while the patient is in the moving stage. So encapsulation enables controlled diffusion, governed by concentration gradients and matrix permeability. So particularly, sir, please change to the next slide. So coming on the industrial scalability. So what we have done, initially we combine the polymeric matrix as well as the cross-linking agent, then we degas. So it is important, degassing is one of the important aspects, because what happens if there are the voids present inside the material. So what are the chances ki like verse release is not possible. So when we are developing it on an industrial scale, so polymeric matrix mixing is important, then the degassing is subsequently is more important when we talk about the encapsulation. Apart than that, we focused on the oil molding and ultimately the further packaging and import and further the quality control aspect of the production. Please change it. So coming on the preclinical material testing, so since this is a bearable drug device, combining product evaluation must include mechanical, chemical, and biological safety. So we focused on all the three aspects of the mechanical safety, chemical safety, and biological safety. So in case of mechanical safety, we focused on the hardness testing as well as the load endurance. In case of drug release, we focused on the release kinetics using front diffusion cell and the release model. So particularly we use the Higuchi model for the drug release kinetics in the development of these insoles. So the model release kinetics to ensure therapeutic but non-toxic exposure is one of the important aspects before we will proceed on the in vivo modelings. So sir, please change. So coming on the in vivo study design, and this is a unique one. Our diabetic rat model was selected. Initially, we have selected the diabetic rat model to evaluate therapeutic efficacy. Please. So sorry for the interruption. So coming on the in vivo study design, we choose a diabetic rat model, and it includes, we particularly focused on the ulcer reduction, epithelialization, microbial load, histopathology, and systematic toxicology. So the entire aim of in vivo study plan is to aim not only on the wound closure, what we focus not only on the wound closure, wound healing, but also the restoration of the tissue architecture is an important aspect. So along with the healing of the wound, we also focused on the restoration of the tissue architecture, particularly in our studies. Please, sir, change it. So just skip it, sir. Yeah, next, skip, sir. So coming on the main aspect, that is the patentability. So unlike existing insoles, as I told you, the existing insoles basically focus on the mechanistic aspect and the improvement of the gait cycle. Existing insoles are individually not having any sort of, they are just having an coating of the antimicrobial coating over it, but there is not any concept of the drug release inside the existing insoles. So, and which provide only cushioning and antimicrobial coating. Our unique device is a delivery system which release the sustained pharmacological therapy through material embedded infusion. Sir, please change it. Please skip it, sir. So coming on he is our co-PI in this project, and this particular aspect is covered by Professor Dr. Khurshid Ahmed Ansari. He is the MD Medicine and Head of the Department of Anatomy at the Jamia Hamdard University in New Delhi. So particularly the device qualifies as a low-risk class B medical device and can be integrated into preventive diabetic footwear system along with the traditional system of drug delivery which we are choosing. So our long-term goal is the development of therapeutic footwear capable of preventing ulcers rather than treating complications. So in concludability, sir, please change the slide. Sir, please skip to the last slide. Last slide. That's a thanks slide. Okay. Please, sir, change. Okay. So in concludability, we prevent a bearable therapeutic platform combining traditional medicine and modern biomedical engineering by integrating sustained drug delivery with pressure redistribution. The device addresses the root cause of diabetic foot pathology, infection, inflammation, and mechanical stress simultaneously. So our work demonstrates how traditional knowledge can be transformed into evidence-based medical technology through scientific validation and engineering design. So this is what a brief glimpse of my research. And if there is any question and suggestions particularly from the audience, please, sir.
Your things is supporting from the wound healing and enhancing the anti-inflammatory enhancing. Okay, fine. But how do you this mechanism compare with conventional pharmaceutical models of the diabetic to wound repair? Fine, sir. So what I am getting, so I have understood. My question is, yes, sir. How do these mechanisms comparing with the conventional pharmaceutical models of the diabetic wound repairs? Fine. So how I am going to compare it? Yeah, that is because that is the support is needed for the to tell the patients. Exactly. So what happened? There is a unique combination which is already existing in the Unani combination. Have you compared for your drug to that conventional? That was that particular drug which is already available there. It is known as Dermatology. It is a combination. It is already there in the market. And that particular combination has been developed by Professor Khurshid only. So we developed some topical formulation out of it. So what we done, sir, we incorporate that particular formulation, ingredient of that particular combination inside the instruments. So what happens is the change is only in the formulation. And we are comparing it with the red models. Because when we come on the clinical models, then actually we can compare it with the existing and approved topical formulation. Whose active ingredient is already there in the system. This is what we use. Sir, any other suggestions? Very good. Thank you.
Thank you, Dr. Navneet. Now we have a talk from Dr. Nitin Sharma, Head of the Department and Professor, MIT Institute of Pharmacy, MIT University, Noida. Dr. Nitin, please.
Thank you very much for this opportunity. I am Dr. Nitin Sharma, working as a professor in MIT Institute of Pharmacy, Noida. And my work is basically on the conversion of this the Unani formulation to the specific colon tablet. So let me discuss the why this is the requirement. So the reason behind it, I am working on the IBS and the IBD. So this is something, the disease which are really existing on some specific location of the GIT. And when we are talking about the targeting, it means that we don't need to send the drug to the entire body. The disease are existing in a specific area, and in my case, the area is the small intestine and the colon. So whatever the delivery system we were using, that was not specifically going to the site, because there was a premature absorption into the stomach. And once the drug are absorbing through the stomach, so it is not going to the small intestine and not showing the better therapeutic value. So the concept of my project were to deliver the drug Jawari Shahi, which was used as a therapeutic substance in case of the IBS and the IBD. So we are incorporating this drug into the colon-specific tablet. And since the drug consists of the herbal product, so there was a requirement to develop a herbal coating material as well. So we convert the Ablomoscus esculentus, which is called normally in the Hindi, this is the bhindi, and in English it is called the okra. When we cut the okra, there is some mucilaginous substance. So we extract out this substance, we modified this substance, and make it as a pH-dependent polymer. We made the conventional tablet of Jawari Shahi. We coat this tablet with the polysaccharide, and which was the pH-specific tablet. Now this tablet will only release the drug to the small intestine and the colon. So this was the general concept. We amalgamate the concept of the beautiful Unani formulation with the traditional methodology of the targeting approach. So this was my work. Can we have the next slide? Next slide. No, it's not moving, sir. Yes. Thank you. Previous slide, please. Okay. Let me tell you what is the IBS. So this is the combination of some symptoms in the GIT. These symptoms are involved the diarrhea, the constipation. Sometimes the patients are feeling the abdominal pain, the bloating, and the main thing is the anxiety. Have you ever felt when you are feeling the anxious, so at the same time your stomach is getting upset? So this is the earlier symptoms of the IBS. And the current Jawari Shahi formulation, next slide please. This is the current, next slide. Sir, you are ready with the presentation? No? Shall I start? Okay. So currently the Jawari Shahi, sir, next. This is the herbal constituents present in the Jawari Shahi. So it's contain the Embellica officinalis, which is normally called the amla. It's contain the Termelia chabula, which is called the harad. It is called the cardamom, which is called the dhania in Hindi. The elaichi, the coriander, and the Salix caprea. So these are the 5 phytoconstituents or the herbal material present in the Jawari Shahi. But the major limitation, next slide please. But the major limitation of the current form is during the administration of the dose. Right? So we cannot measure. Sir, previous slide. we cannot measure the exact quantity, what amount of the drug is required for the therapeutic value. Sometimes the measurable quantity are large, but sometimes the patients are not carrying the minimum therapeutic content. And in addition to this, and in addition to this, there are 50 and to 60% sugar content in the current formulation. This formulation is suitable for the diabetic patients. So my target was to reduce this sugar content from the current formulation. And the major disadvantage was their premature absorption through the stomach. So the entire drug is absorbing through the stomach, and the very least quantity is going into the small intestine and the colon, where the drug is actually required. All right? So this whole combination was making the less therapeutic effect into the disease. So this was the purpose. So we took this conventional Jawari Shahi, we convert into the colon-specific tablet, and this tablet was specifically deliver the drug Jawari Shahi into the colonic area. Next. So while administering, this is the movement of the tablet into the stomach. Next slide. Now the drug is, or the tablet is going into the small intestine, but still the tablet is remain intact. Now it is going into the colon. This is the ileocecal junction. And when the tablet is going into the colon, immediately it will release all the drug into the colonic area. And this was but the purpose, exact purpose of the development of this Jawari Shahi into the colon-specific tablet. So we cannot limit it to this. We can convert any kind of the drug or any kind of the herbal formulation into this concept where the drug is actually required into the small intestine or into the colon. Now, coming to the methodology part, we extract the Abelomuscus esculentus, and after the extraction, next slide please. So these were some methodology. I will show you the data. Next slide. So yes, so this is the okra. It is well-known product, edible product into our kitchen. So we extract out the polysaccharide out of this okra. So this was the yield. Now, this okra polysaccharide was showing the broad range of the pH solubility. So it was swelling at the pH 5 and equally getting the swell at the colonic pH, which was around 7.4. So my concept was to narrow down this pH. So we add the acrylamide in this polysaccharide. This was the chemical process. Next slide. And this is the setup of the acrylamide addition into the polysaccharide backbone. So through this process, we convert this polysaccharide specific. Now this polysaccharide will getting absorbed, the swell at the specific 7.4 pH. So these were some FTIR for the confirmation whether the acrylamide derivative are incorporated into the polysaccharide backbone, and this was the confirmation through the NMR. Next, please. So we performed the DSC just to find out whether what is the characteristic of the polysaccharide before and after, and these were the pulsatile swelling behavior. We incubate the tablet of this polysaccharide. Now, at this time, we are not incorporating the Jawari Shahi, which was my therapeutic substance. This is the only polysaccharide. So this tablet was incubated at the acidic pH, which was the 1.2, and at the same time, this was the incubated at the 7.4. Next slide. And these were the results. So this green line is indicating this is my converted polysaccharide, and the red line is indicating this was the raw polysaccharide of the okra. And the pulsatile swelling study is clearly indicating that when we are keeping or incubating the polysaccharide tablet at the pH 4, which is resembling to the stomach pH, there was the weight was increasing, and it was immediately decreased when we are keeping to the pH 1.2. Then at the 7.4, again there was the swelling. Then immediately there was the de-swelling. So the swelling, de-swelling, swelling, de-swelling. This was the very beautiful characteristic of the converted polysaccharide. And the confirmation of the porosity of the polysaccharide was done with the help of the scanning electron microscope. So this is the raw polysaccharide, and the second image is of the converted polysaccharide. And both the polysaccharide was kept on the 7.4. We can have the very beautiful pores and the porosity in case of the converted polysaccharide, but the porosity was not at that level. Why the porosity is important? Because once the pores will be there in the polysaccharide, that will allow the drug release at that specific pH, right? Next slide, please. Just to confirm whether this polysaccharide was known cytotoxic to the specific cell. So we have this colon cancer cell HCT116, and we find out that at every time, means there was a change in the concentration of the polysaccharide, and every time this polysaccharide was safer, the non-toxic to the selected cell line. So we can go ahead with this data. Then finally, we convert the Jawari-Shahi into the suitable, the colon-specific tablet. This was the flow chart. We convert the tablet formulation. Now this formulation is ready for their preclinical data. How it is in terms of the toxicity and in terms of the efficacy study, we are developing the TNBS-induced the IBS model, and we are finding how, in comparison to the conventional tablet, how effective or how beneficial by tablet is in comparison to the conventional tablet. So these are some additional benefit, and these are the few publications out of these presentations. Next slide, please. So these are the team which are working in this project specifically. She is the Maitri Ghosh. She is my PhD scholar, and the Pradesh Srivastava. And these are the PG students. So thank you very much for the wonderful opportunity. And if any one of you are having any questions, so I would be very happy.
Yes, it's open for discussion. We invite.
Thank you.
So,
Right, sir.
what are the challenges you face with the microflora and pH, and how these strategies influence the efficacy and safety compared to non-targeting deliveries?
Definitely, sir. I am directly coming to their second.
This data. Another thing, why you have taken about the electronic scanning slide?
Okay. So
you have to take from whatever the polysaccharide that may be used, mentioning the name.
Yes.
That may be good for your publication.
exactly.
That is the only my comment.
Right. Thank you.
Yes,
So
good.
I am giving you the answer.
Good suggestions from
Yes.
doctor.
I think, see, your microflora incorporation, definitely this will be going to help in the project, because this is the polysaccharide. And once we are consuming the polysaccharide, definitely it is going to be digested by the microflora which are available into the colon. And in addition to the pH-dependent release profile, we are going to have the microflora-dependent release profile from the drug. So this is a good suggestion, and I will incorporate in the project work. Definitely. Thank you very much. Sorry, sir, dose? Right. Sir, the current dose of the Jawari-Shahi is currently which we are using, that is the 1.6 milligram. So on the basis of that currently available from the dose, we are just converting. However, this was difficult to make the 1.6 milligram of the 1.6 gram of the tablet, so we divided this whole dose into the 2 equal dose of approximate the 800 milligram dose of the Jawari-Shahi.
And most important for Bigari or Amit University, you have all the facilities. Before your formulation, what is the polysaccharide content? After making the formulation, it is there in the formulation or not there, that if you are putting that evidence, so it is very good
Right,
for
right.
the benefit of our community.
Yes.
My simple question is that a number of drugs are available in AYUSH system in specific analysis, but there are concentrations available. What was the rationale behind the selection of the 7 master molecules and a number of drugs? So what was the rationale behind that very carefully?
Right, sir. Sir, rationale behind the selection of this only polysaccharide was, since the drug is of the herbal material, so although in the synthetic material there were the various choice, I can use the Eudragit, I can use the cellulose acetate phthalate, they are already being used for the colon-specific delivery system. But since this is a herbal product, so my motive of using the Ablomoscus esculentus was in Unani system, whether this will be acceptable or not. So that's why I selected the Ablomoscus esculentus. Yes, yes. The plant authentication was there, and there was some set of the experiment before the selection. Yes, yes. There was the Ablomoscus moscatus as well. There was another the species of the Ablomoscus, but specifically I selected the Ablomoscus esculentus, and this species
Both the institutes have a state-of-art facility for preclinical studies, and both are registered and approved by the CCSCA. Next slide. In both the facilities, we used to conduct the toxicity studies of different durations, as well as safety studies of these traditional Unani formulations. And also, we are also running a postgraduate program in both the institute and the Department of Ilmul Adviya, where the students used to conduct their dissertation works in these facilities. Next slide. This slide gives you a brief idea about the preclinical research, which is a link between the basic research and the first used in human trials. So here in preclinical research, we have to develop the proof of the concept, pharmacokinetic profile of the drug candidate, as well as the mechanism of action. If we talk about the pharmacokinetic, we used to evaluate the absorption, distribution, metabolism, excretion, their target engagement in terms of efficacy, which target is involved during efficacy, any kind of side effects or off-target effects of the drugs which will be evaluated during preclinical studies. Third, it is mandatory by the regulatory body that we have to submit the data of the preclinical studies to taking approval for marketing permission of the new drug molecules. However, in case of traditional Unani medicine, we are doing the research in a reverse pharmacology mode, where the efficacy is already established and the people are using these formulations, but we have to generate the evidence of their safety. That's why we are conducting toxicity studies to generate the data. Next slide. This slide is very interesting. This slide is just sharing that how the regulations were developed for animal testing. So in 1937, there was a disaster that is called as sulfenilamide disaster, which was happened in USA, in which by using that sulfenilamide disaster, because that was approved in the market before any toxicity testing, and by consuming it, the 100 people were died. After that, US FDA bring Food, Drug and Cosmetic Act in 1938, which mandate the requirement of the preclinical toxicity testing in animals before getting any drug into the market. Did FDCA 1938 in FDCA 1997, that is the FDA Modernization Act, where the rules were made more stringent to get the approval of the drugs. Now, in 2022, FDA Modernization Act 2.0 comes into the picture, which basically gives you a direction that if we have non-animal model for evaluating toxicity and efficacy, then we should not use any kind of animal. And the reason and the concern because why these guidelines comes into the picture or adopt, the reason which will be discussed in the next slide. Next slide, please. These are the reasons why FDA is going to eliminate animal from the research. The first reason was, the issue was the translatability issue. What we are getting in the preclinical research was not happen in clinical trials. And you will surprised to know that 90% of the trial molecule failed in phase 3 trials, even after getting the best result in the preclinical research. That's why the FDA is moving towards the non-animal model. There is no need to use animals. Second was the animal welfare activities. They also raised the concern that why we are abusing the animals only for research. Third was the poor fit for modern drug modalities like monoclonal antibodies and biologics. These are very complex medicine, and studying these kind of medicine in animal is very difficult. Like if we are doing a trials on immunogenicity molecule, it is very difficult to getting this research into translatability into humans. So these are the reasons. And the fourth one is the non-animal models are more cheaper and cost effective, having less time. Next slide. So these two new FDA Modernization Act 2.0 was come into picture because of these two major changes, which authorize the use of non-animal alternative model for drug safety and effectiveness. And the second is remove mandatory animal studies for drug development. Next slide. There are 3 rules of new approach methodologies. One is replace animal use where possible with scientifically valid non-animal methodologies, reduce the number of animals, and refine practice to minimize stress and improve animals. So the thing is that we have to minimize the use of animals for preclinical research. Next slide. These are the key drivers of change already discussed. Next slide. So in silico modeling and computational. This is actually the generation of artificial intelligence and machine learning, which totally changed the landscape of research, whether it is a preclinical or clinical research. Introduction of artificial intelligence boost the research and in a short time. So this in silico modeling, it gives you an idea about the quantitative structural activity relationship of the molecules, how is the pharmacokinetic profile. We can decide whether we will go ahead or not. Go and no-go decision will be taken in silico modeling. So it will reduce the cost of the research as well as time. Next slide. This is a new technology, organ on a chip model. It's like a pen drive. It's having a miniatured platform lined by the different kind of cells using microfluidic technology. And it is connected with a biosensor technology. You can treat these devices with your drug, whether it is a aqueous or suspension form, and these devices are available like an organ, whether it is a heart, liver, kidney. So without using an animal, we can use this organ on a chip model, and this is the advancement of artificial intelligence and machine learning, that we can, it can give you the idea about the pharmacokinetic efficacy, anything we can get from these organ on a chip using. Next slide. Next. This is also, this is called as organoids, which is similar to the organ on a chip, but the difference is that these are derived from the human stem cells and no microfluidic technology used for developing these organoids. So this mimics the architecture and function of a real organ, and it provides a scalable platform for studying drug safety and biology. Next. The advantages are the better physiological relevance. These are more patient-centric, individualized treatment we can design by using these technologies, and personalized medicine. It enhances the personalized medicine. Next slide. This I think you all know humanized animal models, where we used to incorporate a gene into animals, or we can remove an gene from the animals. So these are the humanized animal models. Next. Now, we are also advancing in biomarkers. Where we now the biomarkers which were available in the serum, we used to earlier evaluate those biomarkers for identification or status of a disease. But now we are going towards the integration of omics technology, where we are studying now proteins, metabolites, genome. Next slide. So this is the integration of multi-omics technology, where we used to study the proteins in the patients. These are the new molecules and the new biomarkers, recent biomarkers. So we are going deeper into the cell. We want to check the effect of a drug on these transcriptomes, proteomes, genomes, which gives a better and relevant results in terms of safety and efficacy. Next slide. Now, if we tell you about the regulatory acceptance, FDA and EMA both are encouraging the use of non-animal models for research. And currently, till now, only one molecule which is developed by AI has been marketed and approved by the FDA. And recent policy developed, this was FDA Modernization Act 2.0, have removed the mandatory animal testing. Next slide. These are the challenges. One major challenge is limited biological complexity, which I have already discussed about the immunogenicity, monoclonal antibodies. So many names lack full systemic integration, such as immune, endocrine, and metabolic interaction. Second is standardization and reproducibility remain concerned, particularly for complex model-like organoids where variability in protocols. And third is additionally implementation of names require specialized expertise and infrastructure. to carry out research on these non-alternative models. Next. So the key takeaway is that introduction of NAMS, new approach methodology, reduce the cost and time, and there is a push by the regulatory authority. So in future, it will be really a growth in preclinical research by using these models, and it will be acceptable all over the world by different regulatory agencies. Thank you. Thank you.
Thank you, Dr. Mohammad Uruz. Now it is open for a discussion. Any questions?
He is the Ustad of the Unani Ustad of the government. And some of the people are sitting in the private. So Ustad must to take from the who are working other universities. And other university where the gaps are missing, that is the responsibility of the Ustad. Also, so that is the only one message I want to circulate for all the people. And most important, the preclinical trends that can bridge classical Unani concept, because we are Unani concept people. We are working for the Unani development. And this is the 12th century information. We have not changed anything which are in the classical text. But now we have to trend from the bridge from the modern. We are in now 21st century. So how with the contemporary pharmacology endpoints, and how should your future studies addressing from the standardization and the mechanism elucidation and translational potential, that you have to tell from the young people who are coming from the taking the Unani studies. That is the you elaborate that thing, because you are a ustad for the private universities.
Yes, sir. The thing which I have already discussed with two eminent speakers from MIT University, the major challenge with our formulation, Unani formulation, is the characterization, because the formulations of Unani is polyherbal. So if we go inside the formulation, there are several constituents. So in that case, we are choosing 3 or 4 molecules, and we are going into research in the form of EOI, expression of interest, and now we are doing pharmacokinetic studies. We are doing the different kind of drug delivery. We are also going towards the using the different advancing AI tools. Presently, we are running 2 AI projects. We are developing the inside soles by Dr. Navneet Sharma, and one is we are developing these cups of hijama on a standard parameters by using different kind of pressures. So we in Unani system, with the time, we are integrating with the modern techniques and artificial intelligence. So in the future, definitely we will be at par with the modern medicine.
Thank you. Next speaker, ये है ना? I invite the next speaker, Dr. Azizuddin Khan. Who? Next. अभी कोई नहीं.
ये तो डिस्कशन है, ये तो रिपोर्टर है, बस यहाँ तक है, ये लास्ट स्पीकर है.
Okay. So we can now. Yes. Definitely, this particular session is well informative, and we have learned a lot of things from the researchers, that how we can collaborate the Unani medicine with the modern techniques, and it should have definitely the basis for which the acceptance will be there in the scientific community, because all the almost trials are conducted, formulated, well planned, well discussed, well analyzed, and that is the success of the research. So all this, I definitely congratulate the young research scientists coming from the countries, from all the faculties, and giving sharing their experiences with us. Now I hand over the mic to my 2 esteemed colleagues, Dr. Ahmed sir and Anwar sir.
Assalamu alaikum warahmatullahi wabarakatuh. In this session, all the presentations were highly informative and intellectually enriching. The first presenter, Dr. Nitin Sharma, Head of Department and Professor, Amity Institute of Pharmacy, Amity University, Noida, effectively curated the treatment of irritable bowel syndrome with Unani formulation, presenting a well-integrated and practical perspective. The second lecture by Navneet Sharma, Assistant Professor, Amity Institute of Pharmacy, Amity University, Noida, provided comprehensive scientific insights into Unani medicine infused organic oil-based insoles for diabetic food care, highlighting an innovative and evidence-based approaches. Dr. Muhammad Uruj Saab, Research Officer, Pharmacology, NIUSD, Hyderabad, delivered an informative presentation emphasizing recent research trends in preclinical studies, offering valuable updates on current scientific developments. Thank you.
Assalamu alaikum. At the end of this morning session, I congratulate all three presenters of today's morning session. They have presented very informative and insightful presentations. Thank you very much, and thank you to audience also.
We'll conclude formally with a memento to the speakers, and thank you, memento to the chairpersons also. First of all, I would invite our first speaker, Dr. Nitin Sharma. Dr. Nitin Sharma. And I would request the chairpersons. And I would request the chairpersons to please felicitate him with a memento and a flower. Yeah. नीचे से दे दो, ये ऊपर लेके दे दो. Thank you, Dr. Nitin, for your lecture. It was a very informative lecture. Now I would invite Dr. Navneet Sharma to please receive his memento from the chairs. Dr. Muhammad Uruj. Now I would request Dr. Najibul Hassan Saab, Director NRM SD Hyderabad, to please felicitate our chairperson, Professor Sadiq Bhomia Patel. Thank you, sir, for sparing out time to be with us. Arey, idhar se nahi, udhar se. Inko dekho. I would request Nagaiya sir to please felicitate Dr. Mumtaz Saab. Sir, please. Let us have the privilege of having you with us. We also have with us Munawar Saab, former AYUSH chair for Bangladesh, to felicitate Dr. Anwar. Huh? Okay. Okay. Ah. He was also the former director for Hyderabad, yes. And we can conclude the session with a group photograph. The speakers, please come on stage. Yeah. Jamil?
पीछे गैप है इसलिए गिर रहे हैं लोग
बहुत अपने डेंजरस गैप है उसके बीच में गैप बहुत ही डेंजरस
Hello. 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 Hello.
Speakers for the session, please load your presentations.
No, I have already got one. Here it is there, ma'am. Okay, fine, yeah. Assalamu alaikum warahmatullahi wabarakatuh. No, actually, sir, I don't have paper. This is for anchoring, sir. This only need. This I will take later for my. Sir, how to make it mute? Mute.
Shift here.
Okay, sir. Okay, now. Good afternoon, everyone. Yeah. Shall I start, ma'am?
Please begin with after tea. The session will start after 10 minutes.
Okay. Okay. Attention, everyone. The session will start after 10 minutes. Tea has been arranged. Please join the tea. Yeah, I have arranged 1 and 2. I have copy pasted. It is there. We have a of Chemical Technology, IICT Hyderabad, as one of the chairperson of this scientific session. Dr. Nagaya is an accomplished researcher with over 33 years of experience in natural products research and herbal drug biomarker isolation. After completing his MSc and PhD from Osmania University, he has led several national and international industrial research projects and guided 18 PhD scholars. He has published 131 research papers and holds 3 national patents. He serves in several prestigious scientific roles, including membership in the Single Drug Subcommittee of the Unani Pharmacopoeia Committee, the Executive and Task Force Committees, and the Scientific Advisory Committee of CCRM New Delhi. He is also the chairperson of the Institutional Ethics Committee at NRMSD Hyderabad. Let us warmly welcome Dr. K. Nagaya. I request sir to chair the session. We are privileged to welcome Dr. Munira Moomin, Principal and Professor at SVKM's Dr. Bhanuvan Nanavati College of Pharmacy, Mumbai, and Director of SVKM C.B. Patel Research Center of Chemistry and Biological Sciences, as one of the chairperson of this scientific session. A recipient of the prestigious Nehru Fulbright Professional Excellent Administrator Fellowship 2019, Dr. Moomin has secured major research funding from AICTE, DST, SCRB, BRNS, AYUSH, and RGSTC Maharashtra. Under her dynamic leadership, the Institute achieved NAAC A++ accreditation, NABA D accreditation, and NIRRF ranking of 39, and received the AICTE C2 Best Industry Linked Pharmacy College Award twice. She serves on various academic boards and has published extensively, holding 8 granted patents with 2 more in progress. Let us extend a warm welcome to Dr. Munira Moomin. I request ma'am to chair the session. We are honored to welcome Dr. Najibul Hassan, a distinguished clinical researcher and administration with over 3 decades of experience in Unani medicine. He currently serves as Director of National Research Institute of Unani Medicine for Skin Disorder at Hyderabad. With expertise in dermatology, rheumatology, gastroenterology, and non-communicable diseases, Dr. Hassan has led and collaborated on more than 15 clinical validation studies, including research on psoriasis, arthritis, peptic ulcer, eczema, and other conditions. He has also conducted multicentric and collaborative studies with National Institute of Unani Medicine, NIM, Bangalore. In addition, he is actively involved in postgraduate Unani education and is widely respected for his leadership, research contribution, and administrative excellence. As director, he is leading postgraduate Unani program in the discipline of Molijat and Advya. His professional journey reflects a unique blend of clinical expertise, research excellence, and visionary leadership. Let us warmly welcome Dr. Najibul Hassan. I request sir to chair the session. We now move to our technical presentation. Our first speaker for this session is Dr. Mohammad Nawab, Research Officer Unani, RRM, Kolkata. We are pleased to welcome Dr. Mohammad Nawab, an accomplished physician, researcher, and academician with over 17 years of experience in Unani clinical research and teaching. He currently serves as Research Officer Unani, Scientist 3, and in charge at Regional Research Institute of Unani Medicine, Kolkata, and is also the coordinator of Peripheral Pharmacovigilance Center, Unani, Kolkata. An alumnus of Aligarh Muslim University, Dr. Nawab specializes in clinical research methodology and clinical trials with significant contribution to studies on neurological, dermatological, and metabolic disorders. He has supervised 25 postgraduate dissertations and published 35 research papers, delivered numerous invited lectures, and serves as an associate editor of a journal published by Springer Nature. He has presented 30 research papers and delivered 15 invited lectures at national and international seminars and conferences. Dr. Nawab has received appreciation certification for publishing the best clinical research paper in 2024 and 2025, as well as the best reviewer award in 2023. He has also contributed as a reviewer for more than 20 high impact fact journals, including the Journal of Ethnopharmacology, European Journal of Integrative Medicine, and Journal of Ayurveda and Integrative Medicine, among others. Today, he will be speaking on understanding cerebrasthenia, zophedimag, a rational approach for its assessment and treatment in clinical practice. I invite Dr. Mohammad Nawab to kindly deliver his presentation. can change from this one.
Bismillahirrahmanirrahim. Respected chairpersons on the dais, dignitaries of the dais, research officers, and dear students, Assalamu alaikum. And very good noon. Today my topic is understanding cerebrasthenia, zoful dimag, a rational approach for its assessment, treatment, and clinical practice. I would like to present in this session about cognitive functions and what is about zoful dimag, diagnosis and treatment of zoful dimag in clinical practice, and outcome of a clinical study, which I have conducted as principal investigator at NRIMSD Hyderabad. Let us know first what is the cognitive functions. Cognitive functions there are 6 in total, and they are named as attention, executive functions, or in alternate way plan or sequence of event, language, memory, procedural motor, and social cognition. These are all cognitive functions normally one person perform. I'd like to take one example of whatever daily activities what we do, the role of these cognitive functions are there. If there is any impairment or any disorder in any of these functions, then it will, the person may not be able to perform their daily activities. Just like I am taking one example, just you want to go pray at any time. Daily you have to pray for 5 times namaz. Though you have to plan first that you get attention that you have to perform namaz. Then whatever requirement for namaz is there, that you have to do wuzu, then you have to recognize a particular place, then you have to perform, and what you have to recite Quran during the period. All these need organization. Suppose that if you have attention but you have not planning, then you will do something before perform namaz, then you can do wuzu. This type of disturbance may be there. Suppose that during the prayer you may not be able to remember any surah, then you will not be able to perform your activity of praying. So every activities, this is just one example. You have every activities, like you are cooking a food item, then you have to know what items is required, where you have to cook, and what is the first, second, and third step will be there when you have to add salt, when you have to spices, then the cooking will be performed. In the same way, a number of activities what you do with the help of these cognitive functions, and any impairment may cause impact on daily activities of your life. So impairment of daily activities of life can be, if there is, suppose that if there is any cognitive impairment, but it doesn't impact daily activities, or it may impact, both the condition may be there. That important point of zoful dimag, you have to understand that cognitive impairment of any of the 6 may be there, but it doesn't impacting a person's daily activities. So that situation is differently recognized in clinical condition. That is very important. If it is impacting activities of daily life, then it is called dementia. If it is not impacting, then it is called mild cognitive impairment. What is the importance of this differentiating point? We will come later on. But my topic is Zoful Dimag. What is the literature of Unani system medicine says that it is characterized by impairment in Quwwat-e Hafiza, Quwwat-e Fikr, or Quwwat-e Takhayyul. These are the faculties, the normal brain of an individual function. If there is any impairment, that is defined as Zoful Dimag in Unani literature. We have taken Cerebrasthenia as an equivalent for Zoful Dimag, and it has been decided by a certain committee, but the definition of this clinical condition is it is a syndrome characterized by chronic exhaustion of brain function, presenting as mental fatigue, decreased cognitive capacity, headache, irritability, and insomnia. So all these possibilities may be there in an individual having this cognitive dysfunction. Now we can have an idea that if there is cognitive impairment, then a person may have its impact on its memory, perception, reasoning, and attention, as well as thinking, judgment, and language. All these faculties of an individual may have an impact due to any impairment or disorder of the cognitive functions. What are the etiology that is defined in the literature is the yabusat or dryness of the brain. That is very important part because the humoral concepts are there, and accordingly the main presentation in the person having Zoful Dimag may be yabusat of the brain. And all these are associated factors that may be chronic common cold, may induce Zoful Dimag, brain tumor, paralysis, and psychological factors just like anxiety, stress, depression, and extreme anger. I have given you the understanding what is mild cognitive impairment and why it has importance, because we are aware that the lifespan of an individual is getting increased day by day in the current situation. Yesterday we have presentation, there is certain zone, 5 zones in the all over the world where a person lives for more than 100 years. But at the same time a clinical condition is developing that is called Alzheimer's, and it becomes a problem after age of 85 years. But before developing Alzheimer's, that includes dementia, and I have defined what is dementia. So dementia is also associated with that. Before developing dementia, it has been recognized that the person may have mild cognitive impairment, and that means it is a condition before age of the 60 and above 50. So in this phase, a range of the period that is more than 50 and before 60, there may be person having mild cognitive impairment, and later on it may convert to Alzheimer's. If somehow we may be possible to have any prophylaxis to not develop mild cognitive impairment, or we may delay the development of mild cognitive impairment, then there will be such type of intervention may resolve the issue of developing Alzheimer's or dementia at the after age of 85 years. So if we define mild cognitive impairment, it is also syndrome, and it has been seen that I have given you the idea. dementia, and the programming age is 75 or 80. Just like I have given you the idea that at the age of 60, person may not change to dementia, but at the age of 70, there is 5%. At age of the 75, it is 10%, and at age of 80, it is 15%, and above 85, it is 25% chances to development of. So in such a way, the physician or clinician, if able to recognize the clinical condition of MCI at an early stage and provide intervention or any prophylactic measure, then it will prevent such situation to be happen in near future. Now, what is what I have discussed about cognitive impairment or zoful dimag. The diagnosis, what is the diagnostic criteria? That is also important for a physician to have an understanding. One thing is that is very important, it is diagnosed clinically. And it is better to diagnose clinically because such type of parameters, biomarkers, if you can detect through that will be a late development, late stage of the dementia. Early stage you have to diagnose clinically, and important thing is clinically what a doctor do generally, either take a history or do examination. But here it is very important. In case of mild cognitive impairment, you have to take history. Medical history is very important, where you will get all the clinical presentation. At the time of visiting the patient to the doctor, they may not have any physical sign, or you may not detect through any clinical examination. Just like neurological examination, you have number of tests like higher function test, like cranial nerve examination, and number of systematic examinations in clinical practice are there, but you will not get any finding. That's why I have kept this slide to have your understanding. Medical history, where you will get the patient will complain, I generally forgot something, by giving some person anything like that, or but that type of presentation may you get in the medical history only. Diagnosis and assessment of severity. Right now, you can add further mini mental state examination, one of the questionnaire based assessment criteria for MCI or Cerebrasthenia, and there are a number of biomarkers are available, but I have given you the instruction not to go for the biomarker examination for MCI, not for dementia or Alzheimer's disease. This is something about mini mental state examination scale. This is the scale, questionnaire based scale, and it may be administered by the patient, but it better, there are certain steps are there that doctor should administer or take this score for this assessment of or screening of the mild cognitive impairment. Not mild cognitive, but cognitive impairment, if there is any. It may be in nature of mild, severe, moderate. It is defined in the later slide. Why this scale I have described here? Because there are 6 domains that has been included in this scale to assess the cognitive impairment, and maximum score is 30, and mild cognitive impairment you can decide when the score is between 19 to 23. A person having 24 score, it is normal, or more than 24, it is normal, and accordingly if it is between 10 and 18, it is moderate cognitive impairment, and if less than 10, it is severe cognitive impairment. Definitely Unani system has described therapeutic approach. The purpose of the therapeutic approach is I have already explained you that it will be better if you can diagnose early, then you can prevent it, or at the same time you postpone or delay it, and if possible then you can reverse it to normal. For this purpose there are mode of therapy that has been described. It all includes pharmacotherapy, dietotherapy and regimenal therapy. So lot of formulations are there indicated in authentic pharmacopoeia. I have given you the idea that essential drug list published by Ministry of AYUSH. It also includes a number of drugs, and Unani pharmacopoeia, where you will get the list. These are the certain examples I have given. They are better for the intervention at early stage, either to delay, and which one what can do I cannot explain you, but it is recommended that it is a potent muqawi dimag for zofedimag in the literature of the Unani medicine. For regimenal therapy, I have seen there are the two therapies, Nutul and Zamad. It is important that site has been described for any clinical domain of the cognitive disorders impaired. Then you select the site and you can do this procedure to get the benefit and intervention. Whatever I have given you, the idea what we have an understanding about the zofedimag, I have conducted one clinical study on this line, and the clinical study purpose was to validate the pharmacopoeial formulation Khamira Gauze-Avasada, and definitely it has objective to evaluate the safety as well as efficacy of Khamira Gauze-Avasada formulation. This study site was NRM SD Hyderabad. It was a multi-center single-arm clinical study and conducted between January 2018 to March 2023. It was a registered clinical trial, and formulation I have studied the Khamira Gauze-Avasada given in 10 gram per day for the 6 weeks. And sample size completed in this study was 72 participants. Inclusion criteria: participants of any gender between 19 to 70 years, and we have taken into consideration the symptoms of cognitive impairment, and we included to recruit the participant MMSE score between 10 to 23. Exclusion criteria: all these diabetes mellitus, hypertension, hyperthyroidism, organic neurological disease, vitamin B12 deficiency, and pregnant and lactating women were excluded from this study. For this assessment criteria for efficacy, it is not safety, it is efficacy. The whatever assessment criteria for efficacy were the two, that one is the difference in MMSE score from baseline to post treatment, and second is change in quality of life from baseline to post treatment. The results, this slide shows the results, baseline characteristics of the participant, and important point is the age, that mean age of the participant were 38 plus minus 12 years, and gender male and female 41 to 56.9, and female participants were 31, and mean duration of the disease 3 years plus minus 2.5 years. Next is the result. We have assessed the efficacy in terms of MMSE score and quality of life. So in MMSE score, the percentage of improvement is 28.6. In quality of life, it is 76.5. But both the change in both the parameters were significant. So we can conclude that Unani formulation Khamira Gauze-Avasada was found effective and safe in treatment of zofedimag. Change in MMSE score and quality of life score was clinically and statistically significant. Issue of intolerability and non-compliance was not reported. And this Unani therapeutic approach to Zofuldemarg has a rationale to diagnose and treat in clinical practice. Thank you.
Now section open for the one or two questions. Please.
Thank you, Dr. Nawab, very novel presentation on a role in Unani medicine. मेरा एक सवाल ये कि यूनानी में जो जोफे दिमाग में कौन सी मेन दवाई मालूम होती है? मुझे
Madam, please silence.
सही है ना? और दूसरा ये है कि जहाँ पे आपने डिस्कस किया रेजिमेनल थेरेपी का, तो हिजामा, तदबीर, ज़्यादा एक सवाल है, एक सजेशन। थैंक यू।
देखिए, important point is my presentation doesn't include all the intervention that is effective or indicated in Zofuldemarg. Literature has a lot of, but I have picked one point because like just regimenal therapy I mentioned, because the site has been identified for a particular impairment in a particular faculty. That was attracted to me. If a general terminology in any clinical condition, all the intervention is used, is not having any significant. That's why I have given you the example. And what was the second question? No, submission, I have replied you first. The first question? Yes. I have given you 3 faculties there. And in the first slide, I have given you that there are 6 domains of the cognitive dysfunctions, and any one or more than one can be possible to define that it is clinical condition of Zofuldemarg. It is not particular that in a particular type. Yes, please.
मेरा सवाल नवाब साहब से है, बहुत अच्छी प्रेजेंटेशन और यूनानी के हवाले से भी संजीदा कोशिश है, उमूमन कंप्लीट। मेरा सवाल ये कि इन्होंने जोफे दिमाग का ताल्लुक डिमेंशिया से और अल्जाइमर से जोड़ा, लेकिन जो उन्होंने सब्जेक्ट्स लिए हैं वो 19 से लेकर 60 तक के लिए। तो मेरा ख्याल ये है कि 19 से 60 लेने का या तो कोई जवाब रहा होगा, या मुझे उम्मीद ये थी कि वो 50 से 60 के लोगों को ज़्यादा शामिल करते हैं।
देखिए, yes, it is very relevant question you have raised. It is a protocol developed by the council, number 1 thing. Number 2 is important that why it has been decided, but it is you have seen that duration of the study is more than 5 years, because when we have included MMSE scale, then we have to examine the patient. I have given you the importance of medical history is there for diagnosis of Zofuldemarg, but it is included MMSE scale, then exclusion of the participant become higher. So insincerely, if I say that I will not get any patient which qualify this scale. That's why it a number of you can say that screening for screening I have to get a higher number of the patient to include. One thing important. Second, whatever I told you, my presentation included the study includes the participant in the age group from 19 to 70 years. You have noticed, but what I found during the clinic during the study that not included the age before 40 years. That is very important to answer your question.
Sir, the main one information I want to tell, because the main key objective for the conducting from this innovation evidence of Unani practice, especially for this area, the council want to make a medicine more accessible and popular for the interest. This is the very important, one of the important area where we are getting. The intention is now from the maybe 0 to 1 year, 2 year, 3 year to up to 70 years.
they are only concentrating on sales. So sub भूल जा रहे हैं उस वास्ते we want to give from the motivate educate from the that is the main intention of the council to start this area. Thank you.
So thank you, sir. Thank you all.
Thank you, Dr. Nawab, sir, for that insightful and enlightening presentation. Our next speaker is Professor Khan Mohammad Khaisar, Professor and Head of Department of Tehfoozi Samaji Tib at Anjuman Islam Dr. MIJT Unani Medical College, Mumbai. Professor Khaisar is widely recognized for his work in interpreting classical Unani concepts through modern scientific parameters and for promoting an interdisciplinary approach to Unani medicine. He is an accomplished orator and panelist at numerous national and international forums, particularly in the areas of interdisciplinary approaches to the interpretation of Unani medicine in modern medical science and Unani conceptual philosophy. He has led 3 research projects, guided 11 postgraduate theses, authored 2 books, and published more than 30 peer-reviewed research papers. An engaging and sought-after speaker, he has delivered over 50 expert lectures at national and international platforms. Professor Khaisar also serves as the managing editor of the peer-reviewed journal National Medicals and is associated with several international academic bodies, including the Academic Board of the AFSA International Crisis Intervention Professional Counselors Association USA and the Global Unani Medicine and Research Foundation Chicago USA and the Avicenna Research Center for Unani Medicine Mumbai, India. He will be presenting on Mental Health in Unani Medicine, a classical contemporary perspective. I now invite Professor Khan Mohammad Khaisar to kindly address the gathering. Welcome, sir. Actually, this is different. Excuse me.
So I am really thankful to the audience. So please, they came all the way from the different places. My humble request, don't use cell and maybe you must be what they are telling. If any gaps are there, you can ask any question. So Why I am telling me there is some time is there. So this is my request for the audience. Now sitting there very well. Sir ji, आपके ही बोल रहे हैं.
अभी बेटा ओपन कर दो डायरेक्ट ऐसे.
Yes, sir. I'll keep in pen drive itself, sir. There's different laptops, so the material will go further. I'll delete from that, sir.
Sir rightly mentioned that thank you for careful listening. I think this is the time for collaboration, cross-discipline, cross-institute. When we listen to the speaker, it definitely gives a lot of ideas, and that's where the collaboration will start, and that's where further the central government idea of promoting traditional medicine would definitely take that vision ahead. Thank you.
Shall I, sir? Sure. नहमदुहु व नुसल्ली अला रसूलिहिल करीम अम्मा बाद फ़ाऊज़ु बिल्लाहि मिनश शैतानिर रजीम बिस्मिल्लाहिर रहमानिर रहीम. Mental health in Unani medicine, a classical contemporary perspective. Since my earlier speaker have already mentioned cognitive kind of thing, I just want to continue with this, that literature serves a pool that explore an idea that paves the way to move on further in research. So here, what I have compiled altogether, what has been taught in classical literature of Unani medicine about mental health, and what could be the prospective, and in today's scientific paradigm, what we can do better for the upliftment in Unani medicine in the concerned field. With these, I move on with this disclaimer. The content of the said lecture is a compilation of teaching material referred from different research papers, article and textbook of various authors, is exclusively for the education purpose. The images used of certain patent product of different manufacturers with their key ingredients in example of various disease is also for education purpose and neither for any marketing, advertisement, nor for any personal gain and benefit. With this disclaimer, I would like to start with a brief narration of understanding what exactly Unani medicine is all about. You know, in conventional science, if we see, there are three basic domains: metaphysics, physics, and biophysics. Metaphysics is a domain of philosopher which deal before the existence of the human being, which deals the cosmology, the universe, the ecology, and all those sciences that give an idea how human come into existence. Physics is a domain that deal with the physical parameter of mass, volume, length, breadth, like kind of things. Biophysics, when that mass or that physical character is being instilled, ruh or the living being kind of thing, so that is being discussed under biophysician. In conventional science, the metaphysics is a domain of philosopher. The physics is mainly deal by physicists, and biophysics is deal by the biophysician. But in Unani medicine, if the health and the disease is being addressed and the curative implementation need to be executed, that at par will be deal by the Hakim. I mean to say, a concept of holism. We need to consider the metaphysical aspect, the physical aspect, and as well as biophysical aspect while addressing the disease of the human being. So this is basically the soul of Unani medicine. If we move further, the Unani system of medicine is based on presumption, hypothesis, which was derived from the metaphysical principle and philosophical doctrine. Hence, assumptions are all embodied in most of all description of Unani text and writing, including its basic fundament, concept and principle. Therefore, we can say Unani medicine is the science of principle. We narrate the principle. Kulliyat, it doesn't go, Juziyat is depend upon individual wisdom. It has been assigned, it has been bestowed to the individual Hakim, that whatever based on those principles, you can implement whatever medicine you want to prescribe as that deem fit in that particular principle. Principles are based on qiyas and tajruba, assumption and hypothesis.
These assumptions were validated on logical argument of philosophy, that is analogy, reasoning. The tool of philosophical argument was empiricism and experiences. With these concepts, we can derive that what exactly Unani medicine's picture is: conceptualization, rational inference, and empirical experiences over the period that has been observed by the treating physicians. While in modern medicine, if we see conceptualization, hypothesis formation, experimental testing, and validation. Now, in this era, under banner of CCRUM and other institutions, these things has been taken under for Unani medicine also. So, these literature serve as a pool that paves the way, that gives an idea what to study, what to research, what to identify, and how to go in that accordance. So in this context, I will start my topic with this introduction, that what exactly Dimagi Sehat is described in classical literature of Unani medicine. Dimagi Sehat, mental health in Unani medicine, is a state of health and well-being of brain, spinal cord and nerves, both in their physical and functional form. Dimagi Marziyat, that is mental infirmity, a dysfunctioning state of these organs both in their structural and functional performance. Mental health and psychic health both used synonymously in Unani medicine, which correspond Dimagi and Nafsani Sehat. If we move further, the nervous system is a complex functioning system. At any given time, as many as activities are being carried out in the body, which is an ongoing phenomena throughout the life. There are senses, there are feeling, there are thoughts, there are ideation, cognition, judgment, moment, etc., for which the nature have blessed the brain as many as power, capacity, and quality to fulfill the requirement. Now, the components of nervous system or the brain health in Unani medicine, there are Dimag or the brain, which think, receive signal, analyze, give order, and then experience, memorize it for future functioning. Nukha or spinal cord is a part of brain, is a subordinate of brain, the functioning bridge between the brain and rest of the body. Asab are the functionary of the brain and spinal cord. They receive signal from organs to brain and spinal cord and carry order from brain to organs. There are asab-e hissi, sensory nerves, and asab-e harki as well, motor nerves. Quwwat-e nafsaniya, that is this actual resemble the central nervous system. This is the apex faculty which looks after all the functionary of the nervous system. Now, Quwwat-e nafsaniya is further being divided into three captions. Quwwat-e mudarrika, perceptive or sensory faculty, the power through which the brain perceives. Quwwat-e muharrika, responsive or motor faculty, the power through which it responds, react or perform. Quwwat-e fikr wa tadbir, cognitive or intellectual or planning or mind faculty, the power through which it analyzes. Rather, in between the perception and the response, there is the processing unit. What exactly takes place in the brain is what the Quwwat-e fikr wa tadbir is. Now, as the literature says that Quwwat-e mudarrika is further divided into two categories, Hawas-e Khamsa Zahira, the external receptors, and the Hawas-e Khamsa Batina, the processing unit within the brain itself. So, there are external and internal senses. So, if we see, there are Quwwat-e Basira as a Hawas-e Khamsa Zahira, Quwwat-e Samia, Shamma, Zaika, and Lamesa. If we see the functioning of the Quwwat-e Basira, so what the thing has been seen by the eyes is being recognized internal Hawas in the brain, that is in the processing unit. So what we see is being recognized by the internal center. The color, the talk, the taste, whatever there, the external channels receives is being processed and analyzed, and a conclusory intuition is being generated in the brain itself. And that is why we perform in actual sense. We recognize, we listen, we plan our body in that accordance. Quwate Mohreka, responsive or motor faculty through which we act. So once we recognize the thing is being processed in our brain, our body start movement. So how it takes place? It is the power which become cause for all bodily movement, actual muscular movement, contraction, relaxation, attainment of posture, crying, laughing, etc. The sequence of all these tahrik and harkat are as following. First, the thought originated from the motor activity to be performed. Thereafter, the sequence of movement necessary to perform the overall task is determined. And lastly, the muscular movement themselves are being controlled. So if we see the sequence, so there are 4 sub-powers under Quwate Mohreka, that is Quwate Khayaliya or Wahmiya, that is power of ideation or thinking, then Quwate Shauq, Quwate Azima and Quwate Faila, which actually from where the activity is being performed. If we see Quwate Fikr wa Tadbir, the cognitive, intellectual, planning and mind faculty, which is being further divided into Hisse Mushtarik, Quwate Wahima, Quwate Mutakhayyala, Quwate Hafiza, Quwate Mutasarrifa. This is the actual the real blessing of the Almighty Allah Subhanahu wa Ta'ala, the Mother Nature who has blessed human being, this intellectual faculty through which the human being is being distinguished with other animates articles. So this is the processing unit. Now, how the functioning of Quwate Nafsaniya took place. So if we want to compile all together in a sense that what a nafs is, how a living entity is. So Quwate Nafsaniya plus Quwate Hayvaniya plus Ruh, that is vital split, is equal to become mind. How we can say that? Mind represent a functional state rather than an independent organ. The brain serve as the physical substrate, whereas the mind denote the manifestation of higher function such as consciousness, perception, sensation, cognition, memory and behavior. The brain provides the structure while the mind reflects the activity expressed through it. Both works as the ruh or the soul ignite, brain grows, mind develops over the period as the age progresses, as the experience counts in person's observations. Mind is the state of one's personality. It's his consciousness, perception, sensation, experiences, way of thinking, approach, attitude. It's all there in the mind because mind and body are interconnected and essentially one. The whole human being exists and function at many levels, spiritual, energetic, emotional, subtle as well as physical. And all these levels interface, interpenetrate and influence each other. And that become one's overall personality. That is called the intellect, the style of working, the thinking, in the performance which is full of life experiences, whether it is positive or whether it is negative. We say that the person is very minded, is very intellect. How that comes? That comes through these channels. Now, if we move in context of pathology, how ones get into whether it is dementia, whether it is other mental health disorder, so how it could influence persons. So whatever life experiences are, all those accumulate and become one's memory based on which an individual determined and planned the life. If everything goes on smoothly and positively, it harmonizes the ruh or the soul, as a result of which the ruh nourishes the body well and give an elixir and tranquility to the body. And if things don't work in one's way, the result is disharmony, failure, loss, refusal, rejection, which put a negative impact on turn into adverse or ill health effect that could serve pathology number 1 for brain health disorder, whether it is depression, whether it is anxiety, whether it is insomnia, and so on kind of thing. If we move further, in classical Unani philosophy, the chain of action initiate in one's mind when a spark of idea is... I have to conclude? So this could be the another pathology where we can say now the disease that has been described, that have to be, can we put together whether they are uzvi or structural disorder, whether they are functional disorder, and there are nafsiyati or hysterical psychiatric disorder. Now we see whatever disease that has been described in classical literature of Unani medicine, we can put them into structural, functional, or nafsiyati or hysterical kind of thing. Accordingly, there are formulation, accordingly there are diseases, there are preparation that can be used in these kind of disorder. So what those basic pharmacological action of those products would be, they could be munakkiye dimag, they purify and cleansing agent, they gives strength to the dimag, mukavviya dimag, then there are muharrike dimag, then there are mufarrih dimag, musakkinat, and musakkine alam. So there are just I want to wrap up everything that not need to be elaborate. What munakkiye dimag exactly sense out of ke tankiya dimag karta kya hai, musakkine dimag ka function exactly kya hai, how it works, and it is all there in the thing, but time is not permitting me to elaborate you all those things. So there are exhilarant, there are musakkinat wa munawwimat, there are musakkine alam and analgesic kind of things. Now in contemporary era, where mental retardation, intellectual disorder, autism, Down syndrome, these are the challenges we are roaring the allied medical health sciences, AYUSH sciences, that to have some solution for this roaring disorder of this time. So we can say that we are having n number of product, including Dimagen, including Itrifal Mukavviya Dimag, including Aksire Mukavviya Dimag, including Memoprash. Now see, these are the patent brand of certain manufacturer things, but they are mentioning that this product can help in this last line, hence can be used in mental retardation, autism, dyslexia, mental development. So these are the patent research oriented product. Unani medicine have a solution for it, subject to whether we use, we prescribe, and we collect the data out of it. So these are the product through which we can serve the community for their health challenges and can prove ourselves that we are a research oriented pathy and we can practice at large. So Khamira Gauzama Nambari Jadwaru Salibola is product of that nature. Then Khamira Gauzaba Nambari Jawaharwala, then Habbe Jund, then Asab Plus, Habbe Azbi Khas, Habbe Azbi Nukra. So there are Kurabadani product as well, there are this pharmacopoeial product as well, and there are patent formulation also available in the market that can be prescribed for the suffering humanity for a solution to cope up all these roaring health challenges of today's world. With these things, I conclude my talk that spectrum of mental health is very comprehensive and detailed in Unani medicine. Implication of basic tenet in managing mental health is important. Hence, to understood basic notion of Unani medicine and implement it in practice, so we can have a resolve these health challenges. Thank you for your patience here.
Thank you very much. One message I want to tell for the audience, especially this area. This is the big market in India and abroad. And the Unani want a focus on immuno-boosting adaptogenesis and nutraceutical is the big market all over the world. They have a lot of scope on this area. And because why means the Unani is originally is a live practice. There is no research on the in original up to 18th century. Then now became it is a practical practice. Practical practice required evidence. So that's why this is the very important area. And one question on behalf of, I think anybody is the any question, I will ask one question to this actually. What that council is expecting, rising the mental health burden is rising. There is no doubt at all. How can the Unani psychological principles be integrated into the modern mental health framework work along the modern psychiatric without losing individual temperament based approach. So because Unani depending on temperament, so now to elaborate for the interest of the audience, one simple sentence.
Exactly. By following the principle what has been described, as I haven't time to make you understand what exactly the spectrum of the phenomena munakkiye dimag, okay? Mukavya dimag. So how they give the actual strength, how they purify, how they remove the waste and the morbid accumulate in the brain. So if those principles are being implemented in classical form, so I think that could be a remedial solution for what sir has been asking.
Yeah. That is, whenever the people are playing with the mind game, they will tell the tensions. When they are playing the brain game is will give the solution. Am I correct?
Yes.
So brain game is most important than mind game. That I wanted to give the message.
Thank you.
सर मैं ऐड करना चाहूँगा
Sir, please.
इसका जो मेंटल हेल्थ का इशू है यूनानी मेडिसिन में, वो सिर्फ ड्रग बेस्ड नहीं है. एक बहुत बड़ा एरिया जो हमारे यहाँ अभी मिसिंग है स्पिरिचुअल मेडिसिन को, जो कि स्पिरिचुअल इन द सेंस ये नहीं कि कोई दुआ करते हैं या कोई ताबीज करते हैं, वो काउंसलिंग है बेसिकली. स्पिरिचुअल वेलनेस पर बहुत सारे एंसेस्टर्स ने हमारी किताबें लिखी हैं और वो सब काउंसलिंग बेस्ड हैं और वो सब के सब टेम्परामेंट बेस्ड हैं. एक आदमी अगर सौदावी मिजाज का आएगा, उसकी काउंसलिंग जो है फिजिशियन अपनी बुनियाद पर करता है. अगर वो सौदावी मिजाज का आएगा तो दवा के साथ साथ काउंसलिंग करेगा. ये बहुत बड़ा एरिया मिसिंग है जो अभी स्पिरिचुअल मेडिसिन के नाम से हमारे पास है. स्पिरिचुअल से उन्मूलन जो है वो मैसेज चला जाता है कि जैसे हम कुछ जादू टोना कर रहे हैं, लेकिन हमारे यहाँ इस तरह का कसूर नहीं है. हमारे यहाँ काउंसलिंग बेस्ड चीज़ें जो है स्पिरिचुअल मेडिसिन में आती है, जो कि मेंटल हेल्थ के लिए हमारे यहाँ सबसे इंपॉर्टेंट है, ड्रग से बहुत पहले. तो ये पूरा एक वो बनता है, जो दायरा बनता है. उन्होंने दवा के हवाले से बात की. मैंने
In my earlier slide, I have already mentioned while addressing any such disease, the concept of holism, all those three domains, whether it is, health has a big dimensions, whether it is mental, physical, social, spiritual, vocational, there are as many as dimensions defined as per the classical format in the WHO. So if we'll consider all those, so it will all include in that. Thank you, sir. Thank you.
So in continuation to this discussion, just one question which came in my mind based on both the presentations, that in Unani practice, what is that one thing, or maybe it cannot be one thing, like how to identify the right mental status, what you mentioned, that there is a gap, right? So probably that could be something to explore, that how do you identify the incorrectness of the temperament, or how to correct that temperament, or how to identify that what is not right, especially with the mental status, mental health, because you have peers, because it is age, right? So if you see peer pressure in young kids, they will not be able to express it well. The same probably the adult would be able to express. So the adulthood or the pediatric depression is something which is very, very important, and probably Unani with a holistic approach can address with the right identification of the temperament in that stage of the age.
जो ह्यूमन बीइंग है, यूनानी मेडिसिन का पूरा कॉन्सेप्ट समझने के लिए 2 मिनट चाहिए. पूरा हम दुनिया में यूनिवर्स से घिरे हुए हैं. यूनिवर्स की हर चीज़ को हमने 4 चीज़ों से जोड़ रखा है, या तो गर्म होगी, या ठंडी होगी, या तर होगी, या ड्राई होगी. अगर जैसे आपने बच्चे के बारे में बताया कि बच्चे का अगर रिस्पॉन्स ऐसा आ रहा है मेंटल हेल्थ के लिए, अगर वो बहुत तेज एक्टिविटी कर रहा है, तो हम उसको हरारत की तरफ यानी हॉटनेस की तरफ ले जाते हैं. ये हॉटनेस की वजह से हो रहा है, और उसको हम फिर नॉर्मलाइज करते हैं हॉटनेस. तो मिजाज का करेक्शन जो हम इन 4 चीज़ों से जोड़ते हैं. कोई नई डिजीज भी हमारे पास अगर कल आती है, जो कि जैसे मान लीजिए कोविड आया, तो हमें उसको पहले उसका ये कैफियत देखनी होती है कि वो गर्म है कि ठंडी है, और किस तरह से मुतासिर कर रही है. और इसी से हम पूरी यूनिवर्स से जुड़ जाते हैं. जो होलिज्म का कॉन्सेप्ट आता है यूनानी मेडिसिन में, वो इन्हीं 4 कैफियत से जुड़ के पूरे यूनिवर्स से हम जुड़े हुए हैं. इवन जो सैयारात हैं, उससे भी हमारा रिलेशन है, वो ऊपर जा रहे हैं, गर्म हो रहे हैं, नीचे आ रहे हैं, उसके भी हमारी किताबों में तस्करे हैं. यानी हम पूरी यूनिवर्स से इन 4 कैफियत के जरिए जुड़ते हैं, और इसी को हम जब बैलेंस करते हैं तो हम नॉर्मल करते हैं. थैंक यू.
Thank you so much, ma'am, for the thought-provoking insights. Thank you, Professor Khan Mohammad Khaisar, sir, for your valuable and enlightening presentation. Our third speaker is Dr. Mohammad Kashif Hussain, Research Officer Botany, S3 at NRMSD Hyderabad, where he leads the Survey of Medicinal Plants Unit. He earned his MPhil 2002 and PhD at 2007 in Botany from Aligarh Muslim University, Aligarh. With over 19 years of research and development experience, he has published more than 50 research papers, more than 1,000 citations, and received several national awards, including the Young Scientist Award 2017 from CCRM and the Best Published Research Paper Award 2025 from the Ministry of AYUSH, presented by Pratap Jadhav and Jitendra Singh. He has held fellowships from the Department of Biotechnology, Council of Scientific and Industrial Research, and National Institute of Plant Genetic Resources, and completed postdoctoral research at the Indian Institute of Integrative Medicine under mentors including the former Vice Chancellor of Jamia Hamdard University. He is also associated with the postgraduate research and teaching program Ilmul Adviyat, NRMSD Hyderabad. He maintains more than 160 germplasm accessions of important medicinal plants of the Indian system of medicine, including Unani, at the Ibn al-Baitar Herbal Garden, providing valuable references material for researchers and students. His current research includes identification and authentication of single drugs used by the Institute's pharmacy unit for clinical research medicine, virtual digitalization of herbarium specimen, and DNA barcode analysis of Unani medicinal plants in collaboration with CSIR CCMB. He also oversees the Institute's museum and herbarium. In addition, he completed the Level 1 to 3rd Professional Training Cum Workshop on Social Media Content Development for AYUSH professionals, sponsored by the Ministry of AYUSH, and actively contributes to AYUSH Unani Social Media Content Development Initiative. He will be speaking on innovative techniques in conservation and sustainable use of medicinal plants. May I now invite Dr. Mohammad Kashif Hussain to deliver his talk.
Sir, I will try to finish in 20 minutes. Just give me 20 minutes and 5-minute discussion. Thank you so much, sir, for inviting me and respected chairpersons, Dr. Nagaya, Dr. Momin, and my director, Dr. Najibul Hassan Saab, and senior research officers, and most importantly, Professor Saeed is here. So I got a lot of motivation from him. And Raza sir is there, ex-in charge, Kabiruddin sir, and all the senior officers, student delegates. All of you know that I have given a topic of innovative techniques for the conservation and sustainable use of the medicinal plants. So whatever they decided to give me the topic related to this conservation and sustainable use. So this innovative techniques, it includes everything, whatever we are doing right now in the age of this artificial intelligence and machine learning. And so it's a very vast topic, and try to cover it in that way that how can we contribute to our system. So I am being a botanist, I am here, and my Professor Idris is also here. Thank you so much, sir, for coming. So I've not seen you, sir. Okay. So the content of my topic will be general introduction for some online live telecast is going on, and the modern crisis, the high-profile Indian species at risk, sustainable development goal, extinction velocity, and what we are doing at CCRM and RMSD, this DNA barcoding authentication, and micropropagation tissue culture, blockchain. And I've taken the case study of this Sandal surk, red sander. It's a Pterocarpus antellina. It's a very important species. It's endemic to Andhra Pradesh region. And one movie has come on that was Pushpa. So why this technique is very important in that case study, and conclusion and some takeaway, if the time permits. So now if you see this AYUSH system, the whole AYUSH system, it's from 11 is it's a transforming to the, it's want to reach at the global level. It's a needs for the globalization, we need certain data in terms of regulatory authorities. So from our ministers and prime minister, this is a message from them that we got, we usually see that from roots to global reach. We are assembled here that whatever we are doing, some take from this, and for policy makers we can give some messages. So this is overall activity of the research activity. Already Dr. Farhat has introduced me that I am working in survey cultivation medicinal plant in Hyderabad. These are the units, and these are the field activities. What we do, that we go to the tribal areas for ethnopharmacological survey and collect the medicinal plants and collect the information from them and document it in that way, that how they are using the medicinal plant, specifically the Unani plants and the Indian system of medicinal plants, and usually we document it and send to the CCRUM. So these are the, you can see the tribal hamlet and how we interact in the different terrain. Even recently I visited the Anantpur Forest Division, and one day on an average we used to walk 7 to 8 kilometers in those areas where the your car or other vehicles can't go. So we in the topic itself, you are seeing that this is a medicinal plant. So how can you define a medicinal plant? So what the plant that cure illness, it has some therapeutic value or pharmacological action, then we can categorize into a medicinal plant. So if you see that outside there is a large number of plants are there and they are ornamental in purpose. Some may be medicinal, but they should have the therapeutic value. So these are the literary thing, and literary persons are here and they are master in that, Dr. Saeed, Dr. Bilal, so all that. So you know that all you are aware that this Discoides describe very effectively about the medicinal plant, and 1,000 recipes have been given in that. If you see this picture, this is very important picture. If you see the map of India, I don't have pointer. So if you see that from India and China itself, there is 10,000 publication. So the reason behind that, in India and China, we are the followers of the traditional system of medicines. Okay, so there is, you can see the number of publication in those areas. So 10,000 publication per year. And you all are aware, because you are mostly from the Unani background, that most of the drugs are come from the plant origin. So why this plant is very important? So we are currently facing triple threat. It's one thing is habitat loss, other is climate change. It's a very vibrant issue nowadays, and unsustainable trade. So how can you describe this habitat loss? Habitat loss means anthropogenic activity, whatever the natural forests are existing, or some way you are disturbing. Even you, yesterday some people have visited the Marine Drive, and you see the extension that below the sea level, there is a transportation system and other things are happening in Mumbai itself. So we are disturbing the natural flora and fauna. This is not the case of Mumbai only. All over India, the same pace is going on. And you are aware that this WHO has given the data that 80% population still depends upon the medicinal plant. And you know that 50 or 70,000 species currently estimated of medicinal aromatic category and are being indiscriminately harvesting at that pace. And you know that 1 in 5 species at risk, and current extinction rates for plants are estimated to be 100 to 1,000 times higher than natural background rates due to anthropogenic pressure. Natural background rates means, if you take the case of the Pterocarpus centellinus, the natural regeneration process itself, it's 10% because of the recalcitrant, 10 to 30%. So that is the naturally occurring. Now we are destroying the plants through the smuggling or the anthropogenic activity or other activities. So some international organizations are very concerned about this, how to conserve these plants. So this IUCN, this International Union for Conservation of Natural and Natural Resources, they have also some estimation all over the world. And you know that the message of our Prime Minister, Honorable Shri Narendra Modi ji, India is a treasure trove of herbal plants. It is in a way of our green gold. So how we are going to defend our green gold? Okay, and how this, everyone is now going for this gold. Now it gone up to the 1,80,000 for 10 grams, and now, so this green gold is very valuable. So now this conservation and sustainability, these are the two things. Conservation is nothing but is a preservation of the biodiversity and the rare species, and sustainability is to ensure the long-term resources availability. Long-term available resources means if we are getting some plants today at that pace, so in future generation after 10 years, 20 years, that should plant sustain. In that way, that plant can be used in our formulations and other AYUSH system of medicine. So the conservation of is two types, in situ and ex situ conservation. In situ means its natural habitat. It means that you are conserving that plant in that way, and ex situ conservation means that outside the natural environment. So the techniques is micropropagation, tissue culture, cryopreservation, other things. These are some basic things. I'm not going to elaborate here. So the economic driver for this conservation or sustainable, that by the year 2025, the industry will go up to the $922 billion. This is a business research insight. This is a latest estimation from that. And so now come to the modern crisis. If you see that the plant of Boswellia, so that has been moved from the vulnerable to endangered category or in critically endangered category due to a perfect storm of over-harvesting, fire, and beetle infestation. Over-harvesting because the pharmaceutical industry are also coming in this natural products, and in a post-COVID era, you can see that a number of companies are producing their formulations on the basis of these medicinal plants. So there is a double loss in that. One loss is that we are using the biochemical library, and other is the secondary metabolite. Nagaya sir is here. He is more concerned about the active constituent. I give a plant to him, Sorelia coralifolia, so he will concentrate only on the sorelin. So if I give him some plant like that, so he will on the so we are losing that biochemical library and secondary metabolites, which are very important. So you know that everyone is now going for ashwagandha. Even Hamdard is making ashwagandha, and they are giving the sample, and everyone is using ashwagandha. So that comes as adaptogen boom. So now where you can get all the ashwagandha, that every pharmaceutical industry will take that. And climate instability is very important. Now you can see that cloud burst and other things are going on, and the CO2 level is enhancing. If it enhances, the sea level will rise, and this is going to affect the population. And habitat for fragmentation, urbanization and agriculture expansion, isolate plant population, and these are the genetic bottlenecks. So weak law enforcement, this is a very important thing, because if you see the red sander or the white sander means Virappan, you all know that we have very weak law enforcement agencies. So we are not implementing the law. Law is still there. The Forest Conservation Act, other acts are there, but we are not implementing it. So this is the report of the Hindu 2022 that 3 Himalayan medicinal plants enter into the International Union of Conservation and Natural Red List. So these are the 3 plants. Now I am coming to this Salam Panja. This is used in our Unani system of medicine. Its current status is endangered. So illegal trade and climate-induced melting of its high-altitude habit, and it's a super herb used for vitality, immunity, and digestive health. And this one is different, Fritillaria chirosa, so I'm not going into detail of that. Now you see, these are the high-profile species at the rate. If you know, I have talked about the ashwagandha, asgan, giloy. Everyone know that whenever you have a dengue fever or you have something else, you can pick the giloy leaf and the papaya leaf, and some pharmaceutical industry are making in that. And sandal suruk, this is important, very important plant, Pterocarpus nataliana. It's endemic to the Chittoor district of Andhra Pradesh. You never get this plant in any area. And the main thing is the powdered wood is used in our system and other things. So a lot of adulteration and other things are going on. So if you see the sustainability goal has been fixed by the WHO. There are 17 sustainability goals. So I'm not going into the detail of that. And of these, one is the life on land. So David Attenborough is very eminent person. Even the student can note down the name of that person, and you can see BBC documentary. So what he told, that IUCN Red List tells us where we ought to be concerned and where the urgent needs are to do something to prevent the despoliation of this world. It is a great agenda of the work of conservationist. So he is a conservationist because. So now if you go to the IUCN site, even you see the status of the Pterocarpus centellinus, there are different category, so it's endangered. There is some assessment process is there. Assessment process, you can submit it to the IUCN data and they will update all this information. Likewise, the Centellum album, sometimes it was vulnerable, now it also comes to endangered category. And one is very important, is the CITES, it's a Convention on Integration of Trade in Endangered Species of Wild Fauna and Flora. So this WWF is going, this is a theme for their, this 2026, that medicinal and aromatic plants, and conserving health, heritage, and livelihood. This is the theme for 2026. So the United Nations medicinal and aromatic plants shines a spotlight on the extraordinary plants that have supported human well-being for centuries. Now CITES is a theme for this 2026. And these are the high-profile other plants. This is Sandal Sufed, Asrol, and Nadosteki Jatamansi. It's Balchad in Unani. And if you see that the status is critically endangered, why it is at risk, they are a different thing. And these 4 plants in Unani system of medicine, it's a Jatamansi, Atish, Kusht, Kutki. So so as the plant become rare, its price increases. Whenever the price increases, like the case of the Pterocarpus centellinus, the illegal poaching, smuggling, and everything is going to happen. So these 3 things are interrelated. So now come to the DNA barcode, and this is a very innovative short DNA sequence technique, and it prevents adulteration and ensure ethical sourcing. If you see the case of the Viola odorata, Banafsha, so some 3 or 4 plants look like the flower of the Viola odorata, but when you dissect the flower and see the internal structure, then only can you identify the Viola odorata. So in that case, DNA barcode is very important. So now you see this is a DNA barcode of this plant, and how it looks like. It's ATCG sequence. Sir, 5 minutes. Why DNA barcode matters? It's preventing adulteration. If it detects the fillers. Fillers means you are getting the saffron-like product in the market, and if you want to purchase the saffron from the market, then you have to go to from the Kashmir or some authorized dealer. And this product is going on, it's collaborative project. Dr. Idris is here from the CCMB CSIR, and we are working on that. And tissue culture, it's a other innovative technique, and it's from the single cell you can produce a number of plants, multiple plants. Even you take the plant and from the suitable media like Murashiji scoop medium or WPM medium, or through this this axillary branching and other methods, nodal there you can get the multiple shoots in the vial itself. It's like a test tube baby. You are getting the test tube baby, test tube plants. Now we are going for the this rooting procedure, and after rooting, the acclimatization. Acclimatization means that from the lab to land condition, and that way this is a very innovative technique. And this innovative techniques even is very useful for the plant like the your red sander. And even for the cell culture, if you are concerned with the person like Dr. Nagaya and other people, even you can synthesize a plant with light, glycyrrhizine, other things. So if for the pharmaceutical industry, going and collecting the plant and harvesting it from the wild resources, even you can use the bioretex and other things. And this is somatic embryogenesis. This is my PhD work, and I did that in Aligarh University, and you can see that how this somatic embryos, even if we can encapsulate it and cryopreserve it, and after some processes we can generate the plant. So this now I am coming to that, AI and satellite monitoring.
If you see the eastern or western Ghat, you have seen, gone through the news of the Tamil Nadu Forest Department that a lot of elephants were crossing the railway track, and now they have AI detector. Even when an elephant is crossing, the driver gets signal that this elephant is crossing the line, and immediately stop the train. So that's how they are managing that forest resources. In Tamil Nadu, it's already been implemented. So for the Eastern Ghat and Western Ghat, we have a microwave high resolution satellite through the ISRO, and even you can, case study of this red sander. I'm going to this IUCN standard status is endangered, and trade is strictly regulated. And you know that hardwood can fetch over 1 crore per ton in the international market. So that's why the picture has come into that. This is a real issue actually. So you can see that this Eastern Ghat, and you can see that how through this satellite and AI and machine learning, everything, the forest, even you can use this RFID smart tag, and it's work and the mesh of network. Then whenever you are picking this, let's suppose this is a population of red sander, I am picking that 3 plants from this one. So that satellite picture RFID will show that these 4 plants has been harvested and posed away. So that's how the technology is working. So access of benefit sharing, this is a social economic issue that innovation has also took place. And what is that 110 crores of rupees has given to the local communities. Whenever you are giving something to the local communities, they will raise the plants, and this harvesting will stop. So the impact of what it did, that 5% of all red sanders auction proceeds go directly back to the local biodiversity committees. Now they are earning from that. So this is the way. So this is the innovation, cultivation and policy, that smart farming, aeroponics, forest to pharmacy, and it's a blockchain is also. Now one thing I would like to share. This financial innovation, that National Medicinal Plant have regional business centers, so even they are spending money on that, and these centers act as a processing hubs where farmers can dry, grade and package rare herbs immediately after harvest, reducing post-harvest loss of 40%. This is a very important issue, that when we are harvesting the plant, whether the active constituent is there or not, the plant is what the plant is. Now this is a legal and regulatory authority where the innovation has took place. This is a report of February 2026 that the supplier have the new digital certificate of origin issued by sharing fee being paid to the local biodiversity management committee. Whatever the benefit is given to the industry, and whatever there should be benefit sharing process is there. So that's why when we are collecting some information from the tribal population or tribal people, we used to get the consent form from there. So these are the legal and regulatory compliances, and now we are in the way that digital Unani pharmacopoeia will come, and this CCRUM is doing validation, and for ethnobotanical survey is the herb source from a CCRUM mapped forest zone. If I am going to some forest zone, whether it was mapped or not, whether I visited the protected tribal land, whether I am going to the Amazon forest and disturbing something, so that should be, can be governed by the AI and other things. So this integrating the Unani industry into the biodigital is going to be a master stroke, and because the single drug mufaradad has been uniquely vulnerable, you know that some plants are very important, very important, and they are endemic. You know that it's very confined to that, and the Unani sector has undergone a massive digital transformation under the CCRUM. Yesterday you have seen the launch of books, the launch of apps, different apps. And I want to leave you with a practical tool. You can have type by, this is not just nice to have. As of February 2026, many of these, like the digital certificate of origin, are becoming mandatory for doing business in the AYUSH and global phytopharmaceutical sectors. If your supply chain is still running on paper logs and vague promises, it's time to upgrade to the biodigital standard. So this is the bridging centuries, 10th century wisdom meets the 2026 precision and bonding. This bridge will come through the CCURM, and the person like Dr. Idris, Professor Saeed, Nagaya, they will do the wonders in there. So combining innovative technologies with traditional knowledge ensures sustainable conservation and use of medicinal plants for future generations. I am very much thankful to our Director General, Nur Zahir Ahmed Saab, DDG Yunus Munshi Saab, my Director Najibul Hassan Saab, Idris Saab, Ghazala Ma'am, GP Pratap, and Anusha Athira, who is still doing the DNA barcoding work. And this is a last slide of thank you, that life on land, this is a sustainable thing, and we have to take sustainability is not an opinion, it is a verifiable data point. Thank you so much.
ये यूनानी का उस्ताद है, यूनानी का उस्ताद, and especially is working in the system. यूनानी का cultivation उस्ताद है. The truth I am telling on behalf of all the members, the novelty coming from traditional and indigenous, there is no doubt at all. And that we want to globalize Unani system for AYUSH as evidence-based medicine. The starting material of identification of authentic raw material is most important. The taxonomy is missing in India. That is the point. Number why means missing in India, with your raw material is wrong means total your formulation is wrong. There is no doubt at all. And most important, the government want. There is in our pharmacopoeias, 1500 species are there. But you take for any institutes, the government or private, whatever, they are only popular using 50 medicinal plant only. They have a lot of scope of young generation. You have to think about other plants which are mentioned our textbooks. That is most important. Finally, I wanted to, because he has to educate from the other institutes. I am asking what scalable innovation model can be developed, linking Unani institutional, local cultivars and biotech labs to shift from the wild collection to climate resilient cultivation while ensuring traceability and preservation of pharmacological active markers. So what is your advice for our audience?
So yeah, if you see my slides, there is a term is aeroponics. In aeroponics, you want to grow the root of Eubedania sonifera, you can get the root there. So why you are going for the wild collection? This is innovative techniques from the Israel and India collaboration. The work is going on. You can do that thing. If you want the leaf of the plant, you can grow the leaf through the tissue culture techniques and other things. For the phytopharmaceutical purpose, you can use the cell lines, cell cultures, and use the bioreactors. Even I visited the Ayurgyan IIT Chennai. During that conference, I visited that IT hub there. So they have a large bioreactors there. So what they are producing, their novel, this active constituent and supplying to the industry. So industry should come forward and give the requirement to the people, and the policy maker will surely do that in that way. And for this conservation purposes or the collection from the wild population, we have to do such thing that we should have the high risk priority plants, and we should go for this innovative techniques, and this way the use of this DNA barcode, and we are collecting the plant of Pterocarpus centellinus, marsupium instead of centellinus. So how you can differentiate in between it? So here the technique comes the DNA barcode. Through DNA barcode, before cutting the plant for whole plant, whole tree, that whole tree comes in the 40 years or 50 years. So now you are cutting the tree, after that you are identifying that, oh, this is not the tree, that tree was Pterocarpus centellinus. So in those cases, you can use these techniques through these innovative technologies.
Okay, thank you. Now fourth speaker. Thank you very much. Congratulations.
Thank you.
So just while the stage is setting up, it's a two-way sword, what Sir mentioned, Dr. Mohammed Kashif. It's a two-way sword. One way we are saying that there is a traditional medicine, evidence-based medicine, we need to adopt modern tools, and that's where we are going for molecular mechanism and pharmacologistic studies. And when we establish these pharmacological studies, the pharmaceutical industries are taking up because they are understanding the importance of traditional medicine and its effect. So one side, we are going for evidence-based microbiology, pharmacology. The pharmaceutical industry is adopting, and that's where the plants are getting endangered. And the other side, what sir rightly mentioned, that is in situ and ex situ, that is test tube, based, tissue culture based. That's where the pharmaceutical industries need to be made aware that instead of going for nature, endangering the plants, they can probably go for some tissue culture or aeroponics, what you talked about. Thank you, sir. It was a wonderful, eye-opening lecture.
It is a collective responsibility. Overall, it's a collective responsibility and collective credits. Yes.
Thank you, sir, for the informative and impactful presentation. Our final speaker for this session is Hakim Misbahuddin Hazar, Research Officer Unani, Scientist for RRM Aligarh. An alumnus of Aligarh Muslim University and Jamia Hamdard, he has over 24 years of research experience and more than 5 years of teaching experience. He has contributed to several clinical research projects, particularly in the area of chronic disease management. He has published more than 100 research papers in reputed national and international journals, presented approximately 60 research papers at various national and international platforms, delivered over 60 invited lectures, and chaired numerous scientific sessions at seminars and conferences. Earlier in his career, he served as senior Unani expert in the joint project Traditional Knowledge Digital Library. His scholarly contributions include Mu'alajat Mushahidat at 2022, Urdu Tibbi Adab Mein Ghair Musallim Atibba Ki Khidmat 2024, and Rubab, a biographical and poetic compilation on Hakim Abdul Wahid Ash Bijnouri. He serves as a member of editorial boards and as a reviewer for more than 15 journals. He is actively associated with several medical societies, serving as General Secretary of the All India Unani Tibbi Congress, Honorary Member Trustee of the International Unani Forum, and President of All India Unani Tibbi Conference. His contributions have been recognized with numerous honors, including the Young Research Award and in 2014, Nishan-e-Ajmal during 2017, Dr. Abdur Razak Award during 2018, Frontline COVID Warrior Award at 2022, National Research Award at 2021, Hakim Ajmal Khan Global Award during 2021, Ibn Sina International Award 2022, and the Dr. C.V. Raman Ratna Award on 2023 for his valuable contributions to Unani medicine and complementary and alternative medicine. He will present on integration of Unani medicine with modern technology in dermatology. I invite Dr. Misbahuddin Hazar sir to kindly deliver his presentation.
Sir, how much time you will give me?
Sir, now time is
It is 1:15. 16.
35, 6 minutes we are telling.
Okay.
1:30
I will try to finish my talk within the time. Okay. So, thank you, Chairperson, and my colleagues who are here, especially Dr. Saeed, Dr. Nighat. My presentation is on integration of Unani medicine with modern technology. Earlier, Unani fellows has used modern technology in terms of the clinical research only for taking the questionnaire-based studies. They have developed by the modern medicines. So they incorporate these questionnaires into our system of medicine, or they use the diagnostic tools. For example, they have used the earlier Unani fellows are using the nerves, ball, bras, and examination, or especially the history taking. They have diagnosed the things. But with the development of the technology, they are using the thermometer, the stethoscope, the bioscope, and everything they have developed. But now I have restricted my talk only related to the dermatology. If we see the objectives of this presentation is that the Unani Hakim have assessed the mizaj and the observations they have taken, and they have suffixed the area of habitat with the drugs also. If we talk about the diagnostic tools, especially in dermatology, we have used the magnifying glass, but now we are using the wood slam, dermoscope, and the KOH mount. But we can use in Unani medicine setup also the direct inflorescence or indirect inflorescence patch test and the photo patch test. Basically, we all are using the smartphones, so we have the facility to click the pictures and send it to the AI, and they diagnose with you, or you can take the margin of that particular area with what is affected and take the things in your slides with the help of the tip of your mobile phone also. If we discuss about the therapeutic tools, so we use the hijama, and hijama is a Unani techniques, but now we have developed a new things just like facial hijama and hijama in other way that is used with the cosmetology. So we didn't discuss about the cosmetological problems. We discussed only the dermatological diseases here only. And the micro needling we are using and phototherapy. So my concern is only integration of the phototherapy along with the Unani medicine in cases of dermatology. So what is the phototherapy? It is a technique that is light based. We are using earlier we using the sunlight, but now we have sophisticated lamps. They are built up with that particular filters that provides the things. Earlier we have instructed to the patient, you apply the medicine and sit on the sunlight, but sometimes it has happened in the big cities or in the area of the weather, we didn't find the sunlight, so we have the complicated these things. So we have developed the things like that. We have applied the medicine and take the help of these developed lamps. If we go to the history, we have find the use of the sunlight in early 400 BC, especially in case of psoriasis. But now we are using the NV-UV therapy in cases of dermatology, especially in vitiligo and psoriasis. If we see the dermatological problem, so according to the modern style of medicine, we can divide these things into a 2-1 that may be a temporary or maybe a permanent. As per the modern medicine, we can say the things psoriasis and the vitiligo in the category of the permanent skin lesions. They didn't be shed out for your whole life, but we can say if you use the modern techniques with the Unani medicine, you can free with these things. So psoriasis, as you know, there may be a different type, maybe a stable plaque psoriasis, maybe a gutted psoriasis, and maybe a erythromatic gutted psoriasis that can be a localized, they can maybe a local. Similarly, in cases of vitiligo, they may be a segmental, they may be a non-segmental, they may be a dermatological, maybe a non-dermatological. We have divided according to the site of the part of the body involved with this one. Earlier, the Unani fellows only say it is a accumulation of bulgum at the particular sites, but the modern medicine say according to their site. So I will try to take first case of psoriasis. You know, the psoriasis is mentioned in Unani literature, that is under 460 to 317 BC by the Hippocrates. And earlier and later on, in early 17th century, the dermatological Robert William recognized the psoriasis and its own condition, and in Glickman in 1986 give the particular name that is taken from the Hippocrates treatises. So in my first case, it was a case of 75-year-old female patient from a Muslim educated family. The patient was non-diabetic, non-hypertensive, but she was the habit of betel chewers as a tobacco and the pan masala from last 30 years. She develops the psoriatic marks in their palm and sore. She was diagnosed in 2004 and taking the medicine from the modern medicine, allopathy, Ayurveda, homeopathy, and sometimes they combined all the things, Unani and homeopathy and Ayurveda and modern medicine. But in 2018 when I was transferred from the headquarters to RRIU Maligal, and I started my clinic in RRIU Maligal. So she came to me and say, I have developed these things from last 10 to 10 years and didn't get the relief. So I have started treatment with the Unani, the Unim 401, that is coined by our former first Director General, Dr. Khalid Siddiqui Saab, who give the name of these drugs. They are the codified. So we use the 2 capsule of 401, 500 milligram twice a day, that contains Fumaria pariflora, Spotia charata, Sorrellia coralifolia, and Terminella chabula in a particular ratio. So, and we have given this in a systemic way, orally, and applied the Sorrellia. It is a marketed product mixed with the clarified butter in a 1:1 ratio, applied on the affected part and exposed to the NV-UV lamp for 10 minutes, and for next day it will be gap, and followed for 15 days up to the 3 months. This was the baseline picture. You can easily recognize the foot of the patient, the right foot, left foot, and both hands. And after 60 days of treatment, there is a remarkable change in their scales and the sensitivity of their foot. And after 90 days, it is totally vanished out kerats at the foot and the hands. Similarly, this was the patient of, it was a mechanic who have developed the demarcated scales on their both hands. He was also the non-diabetic, non-hypertensive, but he was a smoker. He was non-smoker, but he was taking the tambaku in a way, just like a surti. So occasionally oozing out the blood and fluids from the lesion. We have given the same treatment, but topical, but we now change the Dermofresh and as an syrup Entox U. That is also a blood purifier in Unani medicine. The drug, the dosage was given in a way, 20 ml of both the syrup mixed with the equal amount of water twice in a day, and applied the Sorrellia ointment similarly to the case 1. And after 15 days, the picture was this. And after 21 days, this totally vanished out. And both the cases are still in touch with me, and they are passing the 5 years of periods. They are totally cured. So, as a clinician, as a researcher, I can say this is only due to the exposure or the use of the modern technology along with the Unani system of medicine. Just because of that, the drugs is having the property of Musafiyadam, Dafe Tafun, and that is diversion of Akhlaq Salasa. And that drugs helps, but the similar way, earlier Unani treatment was given for the 9 months to 12 months in cases of the psoriasis, but with the use of the NV-UV therapy in cases of the psoriasis, we have get the result in 21 days to 3 months. So with the help of the technology, we can cut down the duration of the treatment. Similarly, it was the case of tinea corporis. A student of 12 standard come to me. I have developed this tinea corporis mark in my hand, and we have applied many things, but it didn't be vanished out. And in the late evening when I was studying, it gave me a trouble for itching and the burning in my hand. So we have given the medicine to him as a blood purifier, Unani blood purifier, and exposed to the sunlight, and sometime it comes to my clinic, so I expose with the NV-UV lamps. After 15 days, the itching was not present, and the scale was present, but on the after 30 days, it is totally cured. And we have follow-up for the next 2 weeks. After 40 to 45 days, there did not be relapses, and she is totally cured. So I am working on today on vitiligo. So vitiligo is an acquired pigmentary disorder with the multifactorial etiology due to the autoimmune destruction in melanocytes. According to Unani philosophy, burrs is developed due to the aggregation of balgame gullies, or there is a diversion of the khilte sauda at the site. So most of the people say it is the accumulation of the balgam, but I say it is the destruction of the sauda. That's why that not be give the resemblance of your skin. So where the balgam is involved, similarly there is the destruction of the sauda also. That gives the color to your skin. If you see the global prevalence of the vitiligo, that is 0.5 to 2%. But in India, it is a very high, 0.46 to 8.8%. In a first case, a female patient of age between 33 years of Hindu Pandit family of Aligarh having the segmental vitiligo spot on right side forehead and near scalp, and the both bottom of her neck. She was belonging to a reputed family, and her father-in-law says, my bahu is not going to outside just because of that they have the vitiligo patch in their forehead and the neck. So please do something. We have started the treatment with the UNIM 001 and 003, that is all, earlier say coined with my earlier Director General Khalid Siddiqui Saab. We have used these medicines for 9th month to 12th month in cases of vitiligo. But now we have started this, the same treatment along with the NVU therapy. So what the result we have got, see in the next picture. Please see the picture. The face was very, just say depressed. Please compare the picture 1 with the 2. The spot has been vanished out, and she has feel the inner feeling of a smileness in his face. So we can say we have not treated the patient, we have spread the smile on the face also. This is the picture. In the second case, a female patient aged between 15 to 20 years from a Muslim family. She was a student of standard 8. She had developed the vitiligo patches in her chest, like this one. At the time of treatment, we have started, and within 1 month of treatment, we get the results. With the UNIM 001, 003, along with the NVU therapy, we have used that modern technology in this area also. In case number 3, a female patient of age between 12 years from the Muslim family of Aligarh having a spot of the neck and a root here. We have we give that same treatment to the patient, UNIM 001, 003. Yes, sir. So we get the results in a 1.5 month. This was the case. One day I was sitting in my clinic. I got the phone from my school days friend, and he says, my partner, he's a builder in Aligarh. My partner has developed some white spot on the face. So I say, suddenly send it to me. He came to my clinic. This was the picture. So I take the history of him. He says, from the last 2 to 3 days, I am developing these patches on my face, but I could not be recognized why they are happening. So I have examined with the wood lamp and say it is a vitiligo. So he's shocked, okay, why I am developing this. When there may be a many cause for that, for the development of this current. There may be a changes in your lifestyle, there may be changes in your diet and things. And I have tried to be convince him, take the treatment from the Unani medicine along with the modern technology, so you will get the benefit. Basically, he is a builder, so he would go to the modern stuff medicine. So I say and convince him, please retain with us for at least 1 month. Inshallah, we will get the results. So see the picture. In a 2.5 months, he is totally cured. So I am trying to finish. So my message is that I say the Unani drugs formulation, as mentioned, our scholars, is effective and safe and need to be used with the precaution and with the confidence in your OPD. And try to be convinced the patient also take the medicine at least for a month or more, not for I say for a 9th month or 12 months, but and you are adding with the modern technology, you will get the results. I will be confident if I try to be pour this confidence to our youngers and the present situation is in front of you. If I am able to give some thing with you, thank you.
Sir, thank you, sir. Thank you. The one message, this present situation, the dermatology problem rising every day due to pollution and our daily food systems is the main cause. And here, this is the only, and our director is sitting here, present CCRA, Hyderabad skin specialist sitting here, and this institute is developed whatever the SAR is telling 001, 003, 004 and 5. They have the lot of clinical data. They have everything is there with that institute. Same time they have approached for the 10 years back to the CSI or IICT. And myself, I contributed for all the markers which are using in 001, 003, 005 and 4 and all this etc. They have already one fellow is trained and he got the PhD in this area. This is the one scope. You can dedicate this formulation to the nation by the support of our DG or the intellectual of Unani intellectual. This is the one scope is there to dedicate nation of this 001, 005 Unani formulation. That I want to say. And sir, one question. How can the institutes like CCRUM and maybe the collaborative with the modern dermatology, because now Prime Minister is asking modern dermatology research centers too, because they're modern, there is no medicine, but Unani there is a medicine. Proudly we can say. And the modern dermatological formulation using omics technologies and clinical validation models. What is your opinion for the interest of the audience?
Sir, you rightly say the director of the NRI SD is here. They are treating lots of in a lack of patients, but they are using the traditional way. Myself is the one person who have developed the
lack of patients.
Yeah, lacks of patients. Lacks of patients. They are their treatment, but they are using the traditional way. Myself is the first person in the council who have developed the phototherapy lab, and I am using these medicine along with the modern technology. I have taken the IMR project from the headquarters. I thanks to my Director General who give the responsibility and trust on me. They have allotted me a project. So this is a few cases we have tried to discuss here. I can say if we are using the other technology in the dermatology, we can inshallah succeed in this area also. Thank you.
It's a lunchtime.
Lunchtime, but you have to tell some comments for the interest of the audience. Sir, because only I will take for the 2 minutes. I am a scientist working in IICT as a phytochemist. I am a phytochemist. I am supporting this Unani system last maybe 18 to 20 years of the Hyderabad. I can proudly told to the in front of the minister, not this present minister, previous minister, and our secretary staff also, how the Unani system is working in India. It is a collective responsibility of the modern people and the Hakims and the modern people. The key objective conducting this innovation and evidence-based Unani practice, this symposium is to make Unani as a modern Unani medicine more accessible to the popular in the country. That is the first our objective. Second objective, strengthen the evidence-based through various research institutes. Already Prime Minister told to the national institutes who are having all the high range equipments, better to, because they are not coming. If I need something, I need some food means I am asking my mother. So your people where the Hakims who are strong Unani formulation, better to approach for the there is 37 institutes in the country, CSR institutes or DSC institutes and DBT institutes. Better to take from the idea, or maybe you can interact, collaboration is must. The third, what we are expecting from this symposium, explore it potential, develop innovative treatment and drugs. That is the most important. And most important, the focus, what we are focusing especially for the council is immuno-boosting adaptogenesis and nutraceutical area, and maybe like ortonics, cardiovascular health, and cosmetic and skincare. This is the big market for the Unani. And most important, aromatic oils and essential oils. Aromatic oil, essential oils, there is a lot of demand. How the Unani system olden days, now what you are adding in the modern. So this is the, why I am telling for the Hakim, this is the one area. And new expedient and of the formulations. Like our fourth speaker told, I am using is not traditional along with the photo. So this point we are expecting. And collaborative research for global outreach means is nothing but we are collaborate. You are the Ustad, CCRM Ustad for the Unani system. But you Ustad, you are interact with not only for India, you interact with China, Japan, South Asian countries who are popularly using from Japan to China. China, they are the little advanced. To Taiwan, you take, Pakistan, you take what? So you have to masters what they are not doing, you have to tell as a Ustad of the Unani system, that collaborate. And most important is the government. The government more interest on the clinical trials. Clinical trials conduct well-controlled, randomized and multicentric clinical trials of whatever the popular, the government expecting not hundreds products. Government expecting from the 5 product from Unani, 5 product from Siddha, 5 product from the Ayurveda. So one product already with you, that is the 001, 003 and 004 and 005. So this is the clinical trials is expecting, and regulator framework of the and resources and resource most important. One thing, whatever the taxonomist told, अभी तक ये प्लांट शॉर्टेज है बोल को कोई पेपर में देखा है क्या? प्लीज रेज योर हैंड्स. ये प्रोडक्ट्स आर शॉर्टेज है बोल को कोई सोचा है क्या? कभी पेपर में न्यूज़ में देखा है? प्लीज नहीं देखा. But government is not allowing, farmers are not cultivating. This question I am telling to the manufacturers, how these people are making without the cultivars are not supplying, government is not owing in the forest. So think it, what they are adding in the traditional system. This is the one. And most important, interdisciplinary research is must for the Unani Hakims, phytochemists, pharmacogens scientists, pharmacologist scientists and toxicology scientists, and framework of the regulations. So these people are really connective. That like-minded people are in the system, in the institutes, in the universities. It will give the fruitful result for the Unani system. So addresses challenges like develop new technologies to stand polyurban pollination, whatever is there, and maybe conduct Q markers are most important for the evidence that you have to take from the, you are not expertise, but you take from the scientists who are working in the CSIR, DBT, DST of the plant phytochemistry area, that you have to take. Addressing the contamination, adulterations, and the maybe heavy metal toxicology, that is the Unani system responsibility. The future direction, what I want to say, conduct larger double blinds, placebo control things, and use omics to other modern techniques to understand mechanism of action, and molecular level, yet understand the mechanism, traditional knowledge, digitization, Akim's text knowledge. We are already insisting that whatever the textbooks mentioned in 12th century to the 18th century, take that you translate in the Urdu, Persian, along with the modern. That is barely needed. You want to globalize your things, and share the research findings with the wider scientific healthcare community to promote accessible healthcare. Then only we will globalize our Unani system. Let work together and revitalize Unani medicine and unlock to full potential. That is, I am as an outsider. I am not a Unani paternity, but I am working for the last 20 years. This is in my mind, because there is a lot of responsibility on pharmacology people. They have to take so many things, so many tests, because only for the getting the plant is only 5%. Working isolation, characterization, and everything is only for the 10%. But remaining your clinical, everything in your hand. And thank you for listening these 4 lectures. I am very thankful. Audience are very sincere and they are not talking. So thank you very much. Thank you very much.
हेलो 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 आ रही आवाज़ जी. थैंक्स टू ऑल डिस्टिंग्विश्ड एंड डायनामिक स्पीकर्स. और बहुत अच्छे आपने काफ़ी अच्छा जो है सब ये करा है. आपका शुक्रिया भी अदा करते हैं हम. और इसके बाद जो है अब तो लंच का बहुत ज़्यादा टाइम ले लूँगा. आपका लंच का भी वक्त हो गया है. अब पेशेंस, थैंक यू फॉर पेशेंस लिसनिंग. थैंक्स टू ऑल. थैंक यू.
Thank you to our esteemed chairperson and distinguished speakers for making this session intellectually enriching and inspiring. On the auspicious moment, we shall now felicitate our esteemed chairperson. As a token of appreciation, I request Dr. K. Nagaya, Senior Principal Scientist, IICT Hyderabad, to kindly present the memento to Dr. Mohammad Nawab, Research Officer Unani, S3, in charge, RRM, Kolkata. Actually, you have to be cautious there. Actually, you go, and this way you go. This way you go, that is better. To give. That's what, so it is not.
Thanks to the organizer giving this hour.
You collect Nagaya sir.
वो पहले देना था मेरे को. It is reverse, उल्टा हो गया.
No, actually, see you. Okay, leave it. So first we are giving for speaker, sir. Actually, it's okay. As a token of appreciation, I request Dr. Jawadul Haq, Research Officer Unani and in charge, CRU Karnool, to kindly present the memento to Professor Khan Mohammad Khaisar, Professor and Head of Department of Tehfuzi Samaji Tib at Anjuman Islam, Dr. MIGT Unani Medical College, Mumbai. You take. As a token of appreciation, I request Dr. Najibul Hassan, Director NRMSD Hyderabad, to kindly present the memento to Dr. Mohammad Kashif Hussain, Research Officer Botany S3 at NRMSD Hyderabad. Najib sir should give the you have As a token of our appreciation, I request Dr. Munira Momin, Principal and Professor SVKM's Dr. Banuban Navanvanti College of Pharmacy, Mumbai, to kindly present the memento to Dr. Misbahuddin Hazar, Research Officer Unani, Scientist for RRM Aligarh. May I kindly invite Dr. Nikat Anjum, Research Officer Unani S4 CCRM New Delhi, to felicitate Dr. K. Nagaya, Senior Principal Scientist, IICT Hyderabad, with flowers and mementos.
So much, sir, for your guidance. That was his moment. Photo he can manage.
More than 200 or 300 mementos are there. What I have to do.
May I kindly invite Dr. Ramesh Manchala, Research Officer Unani and in charge CRU Kerala, to felicitate Dr. Munira Momin, Principal and Professor SVKM's Dr. Banuban Navanvanti College of Pharmacy, Mumbai, with flowers and mementos. May I kindly invite Dr. K. Kabiruddin Ahmad, Deputy Director RRM Chennai, to felicitate Dr. Najibul Hassan, Director NRM SD Hyderabad, with flowers and mementos. With this, we come to the end of the scientific session 6 on recent advancement in Unani research. On behalf of the organizing committee, I thank our distinguished chairpersons, esteemed speakers, and all the participants for making this session intellectually enriching and meaningful. We will now break for the lunch. Thank you all. Have a pleasant lunch. Thank you.
of the Institutional Ethics Committee of Government, Nizamia Tibbia College, Hyderabad. He has published more than 40 research papers in reputed international journals and authored 12 books with special focus on Infialat Nafsania, Mizaj, Umur Tabia, Mualajat Nafsania, Nafsiyat, Mantiq and Falsafa, making significant contribution to classical and contemporary Unani literatures. He is a coordinator of electives under NCISM and has served as a subject expert in multiple selection committee of the Government of India. Let us warmly welcome Dr. Jalil Ahmed, sir. The next chairperson is Professor Basir Lodi, sir, former HOD, Department of Anatomy, Anjuman Islam, Dr. MIJ, Unani Medical College, Mumbai. Professor Basir Lodi, sir, is not here. Because of some reason, Dr. Rashidullah sir is not here. So on behalf of Dr. Rashidullah sir, I invite Dr. Ahmed Saeed sir, Research Officer, Unani Scientist for Hakil Huram, New Delhi. Presently, sir is working as a Research Officer, Unani Scientist for at the Central Council for Research in Unani Medicine, Ministry of AYUSH, Government of India. He is currently involved in literary and historical research related to Unani medicine, served as an eminent expert in the traditional knowledge, digital library, projects of Council of Scientific and Industrial Research, a joint initiative of the Department of ISM and AYUSH Ministry of Health and Family Welfare, Government of India, and CSIR, the project of established with the objective of preserving the classical literature and the national heritage and the traditional knowledge and preventing biopiracy. We truly honored by your gracious presence and academic guidance. Now I invite our first speaker, Dr. Amanullah sir, Research Officer, Unani Scientist Level 4, CCRUM Headquarters, New Delhi. He will be presenting on journey towards the development of Mizaj assessment tools. His work contributes significantly towards objectifying the fundamental Unani concept of Mizaj through scientific methodology. Dr. Amanullah sir is a Research Officer, Unani Scientist 4 at the headquarters of Central Council for Research in Unani Medicine, Ministry of AYUSH, where he is handling a wide range of research assignment and plays a pivotal role in the Council's academic and literary research programs. He is also editor of Council's Urdu Journal, Jahanatib, and associate editor of the English Journal, Hippocratic Journal of Unani Medicine. He has contributed significantly to key research initiatives, including the development of SOPs for Ilaj with Tadbir and development of Mizaj assessment tools. He has published around 60 research papers in national and international journals and has translated, compiled, edited, and vetted more than 20 books on Unani medicine. He has coordinated and supervised over 30 research projects, served on WHO Working Group for Unani terminologies and ICD-11 for traditional medicine. Dr. Amanullah sir has presented papers, delivered lectures, chaired scientific sessions in more than 50 national, international conferences and seminars. I invite Dr. Amanullah sir for his derivation. The stage is yours, sir.
चेंज करना होगा या करेंगे चेंज? वो करेंगे?
No, here, no sir, here you have to change the
ठीक. Assalamu alaikum and very good afternoon to all. It's always a test to have presentation just after lunch in any program. And I can see lack of audience and lack of, I hope a lack of attention would not be here. Lack of audience is somewhat somehow acceptable, but lack of attention is actually discouraging. So I begin my few points regarding journey towards development of Mizaj assessment tool. We all know we are the organizer of this Unani Day and this conference, so we all know that CCRUM is the apex body of for research and development in Unani medicine, not only in India but across the world. I come to the main points. Besides conducting various research that include preclinical research, clinical research, drug standardization research, and survey and cultivation of medicinal plants, and literary research. Besides all these activities, our visionary Director General, Dr. N. Zahir Ahmed, has taken initiatives. Some of them are his initiatives, and we can see some are related to WHO, some are related to Ministry of Health and Family Welfare. So these are some initiatives that have been taken for setting standards in healthcare delivery of Unani medicine. It was discussed yesterday in the inaugural session, and the Honorable Minister of AYUSH and the Secretary as well highlighted the need of having standards in AYUSH systems. So there are, I will be discussing in detail in coming slides, but development of Mizaj assessment tool on which I would be talking is an initiative taken to set standards in Unani system of medicine. So we can see WHO, some projects taken over by WHO and significantly contributed by CCRUM through Ministry of AYUSH to WHO benchmark for practice of Unani medicine, WHO benchmark for the training of Unani medicine, WHO international standard terminologies, and I feel honored here to share that in some of two important documents, WHO international standard terminologies and Unani medicine, our dedicated institute for literary and historical research that is named after Hakim Ajmal Khan Institute for Literary and Historical Research, located in the campus of Jamia Millia Islamia in New Delhi. So officers related to that institute, particularly Dr. Bilal Ahmed, Dr. Saeed Sahab, and myself also, Dr. Miraj Sahab, are associated with these projects, WHO International and ICD-11 also, and that was elaborated yesterday by Secretary of AYUSH. Another project that would be taking many years to complete, ICHI, International Classification of Health Interventions. Oh, sorry. So we means the council through its Institute for Literary and Historical Research contributed significantly in completing these projects. Development of a standard, another step taken by the council to set standards for treatment guidelines and to have a standard protocol for the treatment of for Unani treatment of different diseases. So these two documents were released years back in 2013 and 15 by the council, by CCRUM. These two standards, treatment guidelines have been published by DGHS, Directorate General of Health Services, Ministry of Health and Family Welfare. And the beauty of these documents is the general description related to diseases is common to every system. Every system means allopathy system, Unani, homeopathy, even Ayurveda. And the particular pathophysiology and interventions and treatment protocol have been specified by respective systems. These out of 7 SOPs, 6 were digitally released yesterday. This is a very important step taken over in recent years by our DG sir, and I am very much involved to this project to have standard operating procedures for Ilaj Bi Tadbir. For who are not acquainted with Unani terminology, I would clarify that Ilaj Bi Tadbir is equated in English as regimenal therapy. Regimenal therapy is a very important mode of treatment. There are several modes of treatment in Unani medicine, and Ilaj Bi Tadbir is the first and most important one. So hijama cupping therapy, leech therapy, massage, douche, and fumigation. These important regimens lacked standard operating procedures, and we are taking up this project, and in coming days we would be having more SOPs for different methods of Ilaj Bi Tadbir. Now coming to the main point, mizaj. We all know this word mizaj, aapke mizaj kaise hain? In India we use this to inquire about the well-being of any person. So mizaj, in technical terms, mizaj is the fundamental framework of Unani medicine, and there among few basic principles that have foundational framework of Unani medicine, mizaj is we can say mizaj is the most important and most primary concept. And as we, if you all remember yesterday, short video snippet regarding mizaj assessment tool. So it appeared, it started with a universe, and 4 qualities, hararat, burudat, rutubat, were appearing one by one. So as the concept of Unani medicine suggests everything in the universe, animate or inanimate, has a mizaj that is composed. That may be detailed discussion how it is composed in animate things and how it is composed in animate things, and particularly in humans. Now we come to different sets of observable parameters have been described in Unani medicine to determine or to assess or to know the mizaj of everything. Like if I suggest an example, this microfilm, which is made up of some metals, it also has a mizaj. This wood has a mizaj, and if this flower has also a mizaj, and we humans have mizaj, and that mizaj is not common in all the things. That is particular to everything and to every individual. Mizaj insani, that has English equivalent as human temperament. So this is the idea of my talk. And so mizaj insani is broadly classified into 4 compound mizaj. Like if we see hot and dry temperament, so there may be single kafiyat or single quality also, hot temperament and cold temperament and dry temperament or wet temperament. But commonly it is found in a combination of 2 kafiyat or 2 qualities. So we can say 4 compound kafiyat or 4 compound states of human temperament we can assess as physician and as humans. In this slide we can see if we take up Mizaj as individual kafiyat, then we can have nine parameters or 9 states of Mizaj in humans. So why Mizaj is important for us to adopt measures? In short, there may be more parameters, more points to add for telling the importance of Mizaj, but in short, to adopt measures for preservation and promotion of health, that is one point, and for prevention of diseases, to diagnose disease and to manage disease using various regimens and drugs. Human temperament is assessed by certain bodily, mental and environmental factors. In other words, we can say certain demographic parameters are there, certain environmental factors and certain psychosocial factors are involved in assessing the temperament of a man. Ali ibn Abbas Majusi, a renowned physician of medieval period, has described 5 parameters to assess the Mizaj. We can see. Another landmark concept was given by Ismail Jurjani, another renowned physician of medieval period. Ibn Sina, Avicenna, we must be knowing the name of this legendary person, and at the moment I would like to quote one of the living legend, Professor Padma Shri Professor Sayyid Zillur Rahman, and I remember once he told in a meeting that Ibn Sina, this person, and I would, because I am seeing various non-Unani persons here, so I am focusing on that points. Ibn Sina actually is the person whose books on medicine were taught in European universities till 15th century. And his book, Al-Qanun fi Tib, Canon of Medicine, was an important book of the medical curriculum in European universities. And that book was translated in Latin, French and other European languages. So I was telling that Padma Shri Professor Sayyidul Rahman Sahab, a living legend of Unani medicine, once I heard him saying that if the intellect, if the wisdom has a human state, so that was the state of Ibn Sina. So Ibn Sina told and described 10 parameters to assess human temperament that are listed here in Unani terminology and standardized English terminology. Standardized means that have been standardized by WHO. As I mentioned in my beginning slides, that all the terminologies that are in number more than 4,000 that have been standardized by WHO. So these English equivalents have been taken from that terminology. So actually these 10 features are in vogue even now after the death, after 1,000 years of death of Ibn Sina. Almost all the concepts, basic concepts are given by Ibn Sina through his marvelous books. Development of Mizaj assessment tool, that was that is the initiative of CCRUM. I will be discussing it. So this task, development of Mizaj assessment tool, broad phases. So we have 3 broad phases, and fortunately we are in the completion of 2nd phase. So broad phases, concept development was the sub phase of this phase 1, drafting questionnaire to assess Mizaj assessment, content validation, and statistical analysis. That actually this is not to be said, but being posted at headquarters of CCRM, I was so much busy in taking task related to Unani medicine. Even my associate has prepared this slide, and I was just just saying and instructing him over phone. So this statistical analysis would not have been there. Phase 1 completed, phase 2 pilot study, we are in the completion of this phase, and phase 3 evaluation of accuracy of the tool that would be taken over later. So, because it is related to the basic concept of Unani medicine, Mizaj, and there were many sets, many proformas, many format of assessing Mizaj prevalent in different institutions of Unani medicine. So, first thing was to have a consensus, to have an agreement for how to proceed on this project. So, for that purpose, a workshop that has representation not only from the subject of Unani medicine, particularly the fundamentals of Unani medicine. We had experts from molecular biology, from computer science, from psychology, and I would be discussing why these experts were needed to in the beginning. We can see this in this picture many stalwarts of Unani medicine and other experts chairing on the central place Padma Shri M.A. Wahid Sahab that has not only he has a good knowledge of basic concept, but in translation of basic concept through his laboratories. So Padma Shri M.A. Wahid Sahab, our Director General, and many other professors and experts of Unani medicine. So we had that workshop, a glimpse of that workshop is here. So after having a consensus how to proceed and how to take over this project, the drafting of questionnaire started. And first, as I mentioned earlier, there are 10 parameters to identify to assess Mizaj assessment. Identification of 30 parameters under Ajnase Ashara. Ajnase Ashara means 10 parameters, 10 broad parameters to assess that are Ajnase Ashara, given the name by Ibn Sina. So this is the phase and how we proceeded. This is the questionnaire in final shape. So finalization of questionnaire, there are 10 parameters in the central heading. We can see number 1 in highlighted in blue color, Malmus, and the English equivalent is provided in parenthesis. So under 10 parameters, we have 20 questions drafted, 20 questions to be asked. The 10 domain or 10 broader categories that are in our terminology termed as Ajnase Ashara. So Ajnase Ashara has certain parameters related to demographic like age, gender, and body composition, body state, physique, build, and some are related to the mental state that have been listed in the last, and some are related to environmental factors like infiale aza and what season is comfortable to the subject. So these are the details of 20 questions categorized under 10 broad parameters. So once we finalized 20 questions, and we had various offline meetings, online meetings, telephonic discussions with international experts as well. So after coming to the conclusion of 20 questions to be included in the questionnaire for assessing Misaj assessment tool. Then we had content validity testing for this questionnaire, and for that we selected, not selected, identified 15 experts. Among them, five are international, and we presented questionnaires and we sent questionnaires to them, and on the scale, Likert scale of 4-point Likert scale, relevance, appropriateness and clarity, they scored and they graded all the questions. And after getting their scores, item level content, that is the index for individual category or individual item, ICV, and another we can see a scale level, means or the score of the whole scale. So these were statistically analyzed, reviewed, and critical analysis was done. So on the scale of, on the parameter of relevance, appropriateness and clarity, we got the rating by the experts, and overall rating is 0.82, that is acceptable and significant for these questionnaires. In previous slide, I have deliberately blurred the, because this is raw data and it's not to be shared, it's very early to share all these things. I have to present only how to we proceeded and how its journey started, and how we are moving in this direction. So we got, we had significant ratings from the experts. Now we are in the second phase of the developing the tool for assessing Misaj, and a pilot study in another next slide, to assess the reliability of the Misaj assessment tool on 140 healthy subjects, on 140 healthy participants is designed, and separate protocol is designed, and it was registered under CTRI also. Though there is no intervention, Unani intervention is here, and the study is related, it's only questionnaire based study, and that involve the healthy individuals, healthy participants, but we got CTRI registration also to have this study. And method of assessment, two physicians at same time use this questionnaire in their OPDs, and at baseline they record the data at baseline, at 0 day and at 15 day, and then in intra-rater scoring is done, and inter-rater means a physician between the the physician when completes his assessment at 15 day, so that is also assessed, and the results of the assessment of two physicians is also assessed. So we have initial data on this right now. So, but we have the distribution gender wise and age wise distribution, and in the end we have preliminary data for four Misaj, that are Damvi, Safrawi, Balgami and Sodawi. So I was telling about this inter-rater reliability test, retest, any test and retest by the same physician, and inter-rater reliability between the two physicians. The scoring method applied here explained, and both the ratings, inter-rater and inter-rater, is suggesting significant results. So this is to be traveled to be gone on this in this phase, diagnostic accuracy test. This will be For this phase, we will have involvement of many institutions of Unani medicine, our own institutes and the academic institutions of Unani medicine. On a larger population, we will assess this Mizaj assessment tool to get the diagnostic accuracy, and after that, way forward, finally we would be able to develop a tool that would be accessible to physicians, to even masses, for knowing their Mizaj. So that was some glimpses of our journey towards developing Mizaj assessment tool. Thank you very much.
Thank you, sir, for your insightful deliberation. Now I invite Dr. Fakhre Alam, sir, Research Officer Unani, Scientist Level 4, RRUM, Aligarh. He will speak on contribution of Hakim Ajmal Khan to the rational use of Unani medicine. This presentation highlights historical scholarship and rational therapeutic approaches in Unani practice. Hakim Fakhre Alam, sir, is a distinguished scholar and researcher in Unani medicine. He has been serving as a research officer at the Central Council for Research in Unani Medicine, Ministry of AYUSH, Government of India, since July 2006. He has made significant contribution to the clinical and medical research and preservation of Unani heritage. His specialized domain includes fundamentals of medicine, medical ethics, manuscripts, studies, history of medicine, and biographical literature. His literary output comprising over 15 books and more than 100 research papers published in reputed journals. Some of his well-known books includes Tibbe Mutarjibin, Hindustan ki Tibbi Darsgah, Tibbi Akhlaqiyat, Unani Tib, ka Mughal and Bartanvi Ahad, Rumuze Dawasazi. He has also the esteemed compiler and editor of Razi-e-Hind, Nishan-e-Ajmal and Nuqush-e-Ajmal. Furthermore, he played a pivotal role in the compilation and editing of the standard treatment guideline published by the CCIUM. He has received award from the Uttar Pradesh Urdu Academy and West Bengal Urdu Academy. I invite Dr. Fakhre Alam, sir, for his insightful presentation, and I humbly request all these team speakers to keep the presentation brief in view of the time constraint.
बिस्मिल्लाहिर्रहमानिर्रहीम हामिदुम व मुसल्लिया। मोहतरम चेयरपर्सन्स, रिपोर्टियर, CCRUM नई दिल्ली के यहाँ मेरे बैठे तमाम अहबाब और बहुत ही काबिल एहतराम ऑडियंस, आप सभी की खिदमत में मैं हकीम फ़ख्र आलम सलाम व आदाब पेश करता हूँ। और आज मैं आपके सामने इसलिए हाज़िर हुआ हूँ कि हकीम अजमल ख़ां के उस कंट्रीब्यूशन पर बात कर सकूँ जो उन्होंने यूनानी सिस्टम ऑफ मेडिसिन को रेशनलाइज करने के लिए अंजाम दिया है। एक बात मैं तमहीद के तौर पर जो है अर्ज़ कर दूं कि अक्सर हम मुख्तसर ऑडियंस को देख कर के मायूस होते हैं, लेकिन मैं बिल्कुल मायूस नहीं हूं। इसकी दो वजूहात हैं। इसलिए कि मैं देख रहा हूँ कि यहाँ पर दो तरह के लोग हैं इस वक्त। यूनानी तिब की वो नस्ल या ये वो लोग बैठे हुए हैं जो यूनानी तिब की तश्कील कर रहे हैं, उसको एक शेप देने का काम कर रहे हैं। वो चंद अफराद हो सकते हैं, लेकिन वो अपनी नवीयत के लिहाज से इतने अहम हैं कि वो इस वक्त यूनानी तिब का मुस्तकबिल जो है तय कर रहे हैं। दूसरे मेरी वो ऑडियंस है, मैं जानता हूँ कि शायद आपका ताल्लुक़ बराह रास्त यूनानी तिब से नहीं है, मैंने इसीलिए आपको काबिल एहतराम कहा है। और मैं जानता हूँ कि मैं आज एक उस ऑडियंस से मुखातब हूँ, उससे अपनी बातें पहुँचा रहा हूँ कि जिस तक हकीम अजमल ख़ां की कोशिशों के बारे में, उनके कामों के बारे में जो है उनका जानना बहुत ज़रूरी है। इसलिए कि हम एक आम हलके में, अपने हलके में तो हकीम अजमल ख़ां की बातें करते ही रहते हैं, ऐसे लोगों तक हकीम अजमल ख़ां की बातें पहुँचाना जो कि एक आम हलका ना हो, एक बड़ा ख़ास इस लिहाज से हो कि वो जाएगा तो हकीम अजमल ख़ां की बातें, उनकी फ़िक्र, उनका विज़न या जो उनके थाट्स हैं बहुत दूर तक पहुँचेंगे। तो इसलिए मैं बिल्कुल भी मायूस नहीं हूँ इस ऑडियंस के मुख्तसर होने के बावजूद। तो मैं अपनी गुफ्तगू का आगाज़ इस बात से करना चाहूँगा कि ये मौका यूनानी डे का है। यूनानी डे के मौके पर मुनासिब मालूम होता है कि सबसे पहले आपको यूनानी डे की मुबारकबाद पेश की जाए, उस यूनानी डे की मुबारकबाद पेश की जाए जो हकीम अजमल ख़ां के नाम डेडिकेटेड है। एक बात मैं फिर क़त्ए कलाम करते हुए कह रहा हूँ कि सेमिनार, वर्कशॉप, सिम्पोजियम, कॉन्फ्रेंसेस आप अटेंड करते रहते हैं, हम भी अटेंड करते रहते हैं, ये साल भर होते रहते हैं। एक बात मैं ज़रूर कहना चाहूँगा कि जब यूनानी डे के मौके का अगर कोई सेमिनार होता है तो मैं समझता हूँ कि उसको कुछ आम सेमिनार, आम वर्कशॉप, आम कॉन्फ्रेंसेस से कुछ अलग ज़रूर होना चाहिए कि जिससे हम ये पहचान सकें कि ये जो सेमिनार हो रहा है ये यूनानी डे के मौके पर हो रहा है। और मैं समझता हूँ कि यूनानी डे का सेमिनार और हकीम अजमल ख़ां की बातें अगर ना हों, उनके थाट्स अगर ज़ेर गुफ्तगू ना आए, तो मैं समझता हूँ कि शायद पूरे यूनानी डे का जो मंशा है, जो मकसद है, वो ख़त्म हो जाएगा। तो मेरी 10 मिनट की गुफ्तगू है, लेकिन मैं समझता हूँ कि मैं उस मंशा को पूरा कर रहा हूँ जो यूनानी डे का एक मंशा और एक मकसद होता है। बहरकैफ किससे ज़रा सा आप मुझे वो थोड़ा सा अच्छा यूनानी डे का मौका है, जैसा कि मैंने अभी शुरू में आपसे कहा, तो ये मौका बड़ा अहम है। और इस मौके पर चूँकि हम हकीम अजमल ख़ां को याद कर रहे हैं, हकीम अजमल ख़ां को मुख्तलिफ़ हैसियतों से याद किया जाता है और उनकी शख्सियत की मुख्तलिफ़ जहतें हैं, मुख्तलिफ़ पहलू हैं और उनको खराज अक़ीदत पेश करने के लिए मुख्तलिफ़ अंदाज़ और मुख्तलिफ़ उसलूब अपनाए जाते हैं। अगर मुझे हकीम अजमल ख़ां को खराज अक़ीदत पेश करना हो तो मैं सिर्फ़ और सिर्फ ये बात अर्ज़ करना चाहूँगा कि जिस तरह से आपने देखा कि एक अहद में यूनानी तिब जो इस वक्त कही जा रही है, कभी इसको हिपोक्रेटिक मेडिसिन भी कहते थे, यानी इसको बकराती तिब कहते थे। और एक ज़माने में जब मुस्लिम हुक्मरानों के ज़ेर असर जब भी इस यूनानी तिब का जब नशत सानिया हुआ तो उस वक्त इसको इस्लामी तिब के नाम से भी मौसूम किया गया। लेकिन मैं समझता हूँ कि इस वक्त जो यूनानी तिब है, यूनानी सिस्टम ऑफ मेडिसिन है, उसको अगर हकीम अजमल ख़ां के नाम से अजमली तिब अगर कहा जाए तो ये उसकी बिल्कुल एग्जैक्ट और सही ताबीर होगी। अजमली तिब उसको इसलिए कहा जा सकता है कि इस वक्त यूनानी सिस्टम ऑफ मेडिसिन का कोई भी इस वक्त शोबा या डिपार्टमेंट ऐसा नहीं है, चाहे वो एजुकेशन का हो, चाहे वो रिसर्च का हो या फंडामेंटल्स का हो या जिस तरह का भी शोबा हो, कोई भी शोबा यूनानी तिब पे इस वक्त ऐसा नहीं है जो कि हकीम अजमल ख़ां के ज़िक्र के बग़ैर, कोई भी शोबा ऐसा नहीं है जो कि हकीम अजमल ख़ां के ज़िक्र के बग़ैर उस शोबे की तारीख़ या उस शोबे का ज़िक्र जो है मुकम्मल हो सकता हो। हम मुकम्मल ही नहीं कर सकते कोई भी बात अपनी उस वक्त तक जब तक कि हम हकीम अजमल ख़ां का नाम ना ले लें। हकीम अजमल ख़ां के यूनानी तिब में ज़ाहिर है कि मैंने एक ही बात में वो तमाम बातें समेट दी कि चाहे एजुकेशन का मसला हो, रिसर्च का मसला हो या यूनानी तिब के जितने भी इस वक्त इशूज़ और मसाइल हैं, उसकी फहरिस्त एक बहुत लंबी हो सकती है। मुझे हुक्म हुआ है, अभी इशारा हुआ है कि आप अपनी बातों को ज़रा मुख्तसर ही रखिएगा तो मैं सिर्फ ये बात अर्ज़ करना चाहूँगा कि जितनी भी चीज़ें हैं उस सब में हकीम अजमल खां बरह रास्त शामिल नजर आते हैं, इसीलिए इसको हम जो है, मैंने अपनी गुफ्तगू में ये कहा था कि अगर अजमली तिब कहें तो शायद बेजा ना हो, लेकिन बहुत ज़्यादा तमहीद में और गैर ज़रूरी चीज़ों में अपनी बातों को न ज़ाय करके मैं असल मुद्दे पर और असल मौजू पर आना चाहूँगा। और ख़ास तौर पर मैं आज इस पहलू पर आपके सामने जो है अपनी गुफ्तगू को रखना चाहूँगा कि जो रेशनैलिटी के लिए, जो साइंटिफिक रीज़निंग के लिए जो हकीम अजमल खां ने जो कोशिशें की हैं, जिसका कि मकसद ये था कि आप उसको जो है रेशनलाइज कर सकें, ख़ास तौर पर मैं उन पहलुओं पर अपनी गुफ्तगू को जो है फोकस करने की कोशिश करूँगा। आपको ये पता है कि जब हम कोई बात करते हैं, कोई वार्तालाप करते हैं, कोई जब डिस्कशन करते हैं, तो उस डिस्कशन का, उस गुफ्तगू का एक बुनियादी क्वेश्चन होता है। उस बुनियादी क्वेश्चन को अगर मैं ये कहूँ कि आज मेरी गुफ्तगू का बुनियादी क्वेश्चन है क्या है, तो बुनियादी क्वेश्चन यही है कि उस वक्त जबकि यूनानी तिब के सामने बहुत सारे मसाइल थे, तो उस वक्त रेशनलाइजेशन के लिए हकीम अजमल खां ने यूनानी सिस्टम ऑफ मेडिसिन के लिए क्या रोड मैप बनाया था। बेसिकली यही मेरी गुफ्तगू का आज जो है असल सवाल होगा, और इसी सवाल के इर्द गिर्द मेरी गुफ्तगू जो है आज जो है इंशाअल्लाह गर्दिश करेगी। जब हम रेशनल की बातें करते हैं तो अक्सर हम कहते हैं, लेकिन मैं समझता हूँ कि बहुत सारे लोग एक वाज़ेह सोच अपने अंदर या अपने जहनों में नहीं रखते हैं कि एक्चुअली रेशनल यूज़ या रेशनल का मतलब क्या होता है। जब हम यूनानी सिस्टम ऑफ मेडिसिन के रेफरेंस से कहते हैं, तो रेशनल का बुनियादी जो मकसद होता है वो ये होता है कि नंबर 1 चीज़ ये है कि करेक्ट डायग्नोसिस होनी चाहिए, नंबर 2 ये है कि एप्रोप्रिएट मेडिसिन होनी चाहिए, नंबर 3 ये है कि राइट डोज एंड ड्यूरेशन होना चाहिए, और फिर नंबर 4 जो चीज़ है वो ये है कि सेफ्टी और एफिकेसी का उसमें जो है लिहाज होना चाहिए। जब ये 4 चीज़ें पाई जाएंगी किसी भी चीज़ में, तो उसको हम जो है क्या कहेंगे, ये चीज़ जो है रेशनल है। अब ये मसला ये है कि अगर हकीम अजमल खां ने रेशनलाइजेशन किया है, तो इसका मतलब ये हुआ कि यूनानी सिस्टम ऑफ मेडिसिन इरेशनल था। इरेशनल को दूसरे लफ्ज़ों में यूँ कह सकते हैं कि इलॉजिकल था, गैर मंतकी था, जिसकी कोई बुनियाद नहीं थी। यही तो मतलब हुआ, शायद मेरे ख्याल से यही मतलब हुआ, जो कि बड़ा सख्त होता है। रेशनलाइजेशन की जब हम बात करते हैं तो बहुत अच्छा लगता है, लेकिन उसका जो बैकग्राउंड है और उस बैकग्राउंड में जो एक मीनिंग छुपी हुई है वो बड़ी तल्ख और सख्त होती है। तो अब आप ये बताएं कि जब रेशनल यूज़ की वो डिफिनेशन जो कि अभी इससे पहले मैंने आपके सामने पेश की है, अब उसकी रोशनी में अगर यूनानी तिब को इरेशनल कहा जा रहा था तो उसका मतलब क्या था। जाहिर है कि उसका मतलब यही था कि रेशनल का मतलब क्या था। पहली चीज़ ये है कि उसके लिए हमें जो है करेक्ट डायग्नोसिस होनी चाहिए। जाहिर है कि यूनानी स्कॉलर बगैर डायग्नोसिस तो इलाज नहीं करते थे। दूसरी बात यह है कि वो दवाएं इस्तेमाल कर रहे थे जो हज़ारों साल की एक्सपेरिमेंटेड थी, और जाहिर है कि जो है डोज और ड्यूरेशन का मसला भी उनके यहाँ तय था, और सेफ्टी और एफिकेसी के तमाम पैरामीटर जो उस ज़माने में जो राइज हो सकते थे, इन तमाम बातों को वो लिहाज करने के बावजूद अगर किसी चीज़ को इरेशनल कहा जा रहा था तो उस इरेशनल कहने वाले कौन थे और इरेशनल उसको क्यों कह रहे थे, ये मेरे ख्याल से शायद आज आपका जानना बहुत अहम है। तो बुनियादी तौर पर वो 3 चीज़ें थी जिसकी वजह से यूनानी सिस्टम ऑफ मेडिसिन को वेस्टर्न मेडिसिन के लोगों ने उस वक्त इरेशनल कहा था। पहली वजह क्या थी, उनका ये कहना था कि इसमें जो है थ्योरेटिकल एम्बिग्यूटी पाई जाती है, यानी ये महज तसव्वुराती चीज़ें हैं, गैर मादी चीज़ें हैं, और अगर तसव्वुराती या गैर मादी चीज़ों की जो बुनियाद है इस पैथोफिजियोलॉजी की बुनियाद पर अगर कोई डायग्नोसिस अगर जो है बनाई जाएगी तो वो जाहिर है कि सही नहीं हो सकती है। ये उनका बुनियादी तौर पर एतराज़ था कि इसमें जो है थ्योरेटिकल एम्बिग्यूटी यानी नज़रियाती इबहम पाया जाता है। दूसरा उनका जो है बहुत अहम एक जो है एतराज़ ये था कि लैक ऑफ एम्पिरिकल एविडेंसेस, यानी ये तजर्बाती शवाहिद नहीं रखती। मैं अब तक नहीं समझ पा रहा हूँ और न तो मेरे किसी दोस्त ने मुझको ये समझाया कि तजर्बाती शवाहिद का मतलब क्या होता है। 20 केसेस की स्टडी अगर तजर्बाती शवाहिद रखती है तो 20 लाख केसेस की स्टडी को हम तजर्बाती शवाहिद न रखने वाली आखिर क्यों कह रहे हैं। वाकई मैं इस चीज़ को नहीं समझ सका हूँ और न तो मुझको कोई इत्मीनान बख्श जवाब अब तक इस चीज़ का मुझे मिल सका है कि लैक ऑफ एम्पेरिकल एविडेंसेस। तीसरा उनका जो एतराज़ था वो ये था कि इसमें स्टैंडर्डाइजेशन यानी मेयार की जो है कमी होती है। वो कौन सा मेयार चाह रहे थे, मैं इसको आगे ज़रा सा और वाज़ेह करूँगा। मैं थोड़ा सा बहुत सारी चीज़ों को जो है छोड़ता हुआ जा रहा हूँ, सिर्फ उसकी हेडिंग को पढ़ता हुआ जा रहा हूँ, इसलिए कि मैं जानता हूँ कि यहाँ पर जो है बेसिक चीज़ ये है कि क्या वेस्टर्न मेडिसिन के लोगों ने जो यूनानी सिस्टम ऑफ मेडिसिन को जो सही साहब जो इरेशनल कह रहे थे, तो क्या आप इससे मुत्तफिक हैं? क्या इसको इरेशनल कहा जाना चाहिए? बहुत बुनियादी चीज़ है, मैं हूँ, आप हैं, आप हैं, और आप यहाँ बैठे हुए जो ऑडियंस हैं, मेरे ख्याल से हर इंसान अपनी एक आइडेंटिटी, एक पहचान, एक शनाख्त रखता है। ऐसा नहीं कि सिर्फ इंसान कोई एक शख्स बैठा हुआ हो सकता है, बाकी लोग नहीं हैं। तो है क्या चीज़ कि जब यूनानी सिस्टम ऑफ मेडिसिन और वेस्टर्न सिस्टम ऑफ मेडिसिन जब वो दो अलहदा पैथीज़ हैं, तो जाहिर है कि दोनों के अलग अलग फंडामेंटल्स हैं, दोनों की अलग अलग बुनियादें हैं। तो इल्म व अदब की जो दुनिया होती है उसमें एक दूसरे की पहचान, उसकी आइडेंटिटी और उसकी शनाख्त जो होती है उसका एहतराम किया जाता है, उस पर गुफ्तगू की जाती है। उसको ये नहीं कह सकते कि तुम तो इंसान ही नहीं हो। ये तो वही हुआ कि कोई किसी शख्स को ये कह दे कि तुम तो इंसान ही नहीं हो, तो बिल्कुल जैसे ये कहना कि तुम इंसान ही नहीं हो, वैसे यूनानी सिस्टम ऑफ मेडिसिन को मैं समझता हूँ कि इरेशनल कहना था। इसलिए कि मैं वेस्टर्न सिस्टम ऑफ मेडिसिन को जो एतराज़ था, कम अज़ कम यूनानी तिब के एक स्कॉलर के तौर पर मैं उससे हरगिज़ इत्तेफाक नहीं करता। अब तक हमने क्या देखा? जो मैंने अब तक 5 मिनट, 7 मिनट, 8 मिनट की गुफ्तगू की है, उसमें हमने ये समझने की कोशिश 15 मिनट हो गए। बहरहाल हमने ये देखा कि रेशनल क्या होता है, वेस्टर्न सिस्टम ऑफ मेडिसिन के लोग यूनानी सिस्टम ऑफ मेडिसिन को रेशनल क्यों कह रहे थे, और ये भी हमने देखा कि उनके जो एतराज़ात थे वो किस हद तक दुरुस्त थे। तो अब इसके बाद मैं जो असल मेरी गुफ्तगू का जो महवर था, मैंने कहा कि बुनियादी सवाल मेरी गुफ्तगू का ये होगा कि रेशनलाइजेशन के सिलसिले में हकीम अजमल खां की जो कोशिशें रही हैं वो उन्होंने क्या किया था। तो अगर मेरी पिछली गुफ्तगू आपको याद होगी तो आपके जहन में ये बात होगी कि सबसे पहले उन्होंने ये कहा था कि थ्योरेटिकल एम्बिग्यूटी इसमें पाई जाती है, यानी नज़रियाती इबहम पाया जाता है। तो इसके करेक्शन के लिए हकीम अजमल खां ने जो सबसे पहला काम ये किया कि उन्होंने, हकीम अजमल खां का सबसे बड़ा कंट्रीब्यूशन है कि उन्होंने इंस्टीट्यूशन की शक्ल में यूनानी तिब की एजुकेशन का न सिर्फ इंतज़ाम किया था, बल्कि एक मॉडर्न क्यूरिकुलम और एक मॉडर्न सिलेबस के ज़रिए उन्होंने यूनानी तिब की तालीम का इंतज़ाम किया था। दूसरी चीज़ ये कि मैंने उसको बिल्कुल हाईलाइट कर रखा है कि हकीम अजमल खां का कहना था कि न सिर्फ ये कि एक मॉडर्न क्यूरिकुलम की इस वक्त ज़रूरत है, बल्कि मादरी ज़बान में जो मदर टंग होती है उसमें जो है तालीम की अहमियत को हकीम अजमल खां ने बहुत अच्छी तरह से वादे करने की कोशिश की। जिन लोगों का ताल्लुक़ एजुकेशन वगैरह से है, आजकल जो एजुकेशन वगैरह की जो नई फ़िलासफ़ीज़ आ रही हैं उसमें इस बात को वो कहा जा रहा है कि एजुकेशन जो होनी चाहिए, जो जिस लैंग्वेज का है, जो जिस ज़बान का है, जो जिस भाषा का है, उसकी अपनी मादरी ज़बान में या मदर टंग में होना चाहिए। हकीम अजमल खां का ये कहना था कि आप एक बहुत बड़ा रिसर्चर पैदा कर सकते हो, एक बहुत बड़ा स्कॉलर तो पैदा कर सकते हो, लेकिन अगर मादरी ज़बान में तालीम नहीं हुई तो आप उस सिस्टम या उस फ़न का एक दानिश्वर कभी नहीं पैदा कर सकते। तो मैं समझता हूँ कि इस वक्त जो सबसे बड़ा मसला यूनानी सिस्टम ऑफ मेडिसिन का है कि स्कॉलर तो हमारे पास हैं, लेकिन शायद फ़न दानिश्वरी की जो है मैं समझता हूँ कि अब्दुल जलील साहब इसकी बड़ी कमी महसूस हो रही है। बिल्कुल आपने इत्तेफाक किया इससे। अच्छा, वक्त ख़त्म हो गया? खैर, नहीं, बस कोशिश कर रहा हूँ। एक दूसरा सवाल मैं खाली एक सवाल की शक्ल में उठाकर कि इस बात को यहाँ पर छेड़ करके जाना चाहूँगा कि हकीम अजमल खां ने ये जो मॉडर्न क्यूरिकुलम जो ले आए थे, ये बेसिकली उस ज़माने की एक मोमेंट थी न्यू हिपोक्रेटिज्म या नौबुकरातियत के नाम से। हकीम अजमल खां ने उससे मुतासिर होकर के किया था, और इस मोमेंट के जो फाउंडर थे ये कोई इंडियन नहीं थे, ये सब के सब जो है इसके जुड़े हुए जितने भी लोग थे वो सब के सब यूरोपियन थे, और सब के सब मॉडर्न मेडिकल साइंस के वो न सिर्फ स्कॉलर थे बल्कि बहुत ऊपर के लोग थे। उन्होंने ख़ास तौर पर वेस्टर्न सिस्टम ऑफ मेडिसिन की जो जर्म थ्योरी है और जर्म थ्योरी की जो टारगेटेड अप्रोच का जो एक कॉन्सेप्ट है उसकी मुखालिफत में उन्होंने वो किया था। बहरहाल जर्म थ्योरी, एंटीबायोटिक और ये तमाम चीज़ों के ताल्लुक़ से जो असरात सामने आ रहे हैं, मेरे ख़्याल से न्यू हिपोक्रेटिज्म और नौबुकरातियत के लोगों ने जो चीज़ सोचा था वो चीज़ें तमाम और मुकम्मल सूरत में इस वक्त जो है एलोपैथिक सिस्टम ऑफ मेडिसिन या मॉडर्न वर्ल्ड के सामने बिल्कुल जो है वाज़ेह हैं। दूसरी बात यह है कि मैं एक बात ख़ास तौर पर और कहना चाहूँगा कि उन्होंने ये कहा था कि तुम्हारे यहाँ इम्पेरिकल एविडेंसेस की कमी है, दवाओं के सिलसिले में उन्होंने बात की थी। इस पर मैं बहुत अहम बात करना चाह रहा था, लेकिन ये कि वक्त की कमी की बुनियाद पर सिर्फ ये बता दूं कि उस वक्त हकीम अजमल खान ने, चूँकि रेशनलाइज उनको करना था, तो उन्होंने क्या किया था, ये बड़ी लंबी कहानी है। डॉक्टर सलीम सिद्दीकी और ये वाक़या आप सभी हजरात इससे वाकिफ़ हैं, लेकिन यहाँ पर जो ख़ास तौर पर मेरे तिब के साथी हैं, मैं उनको मुतवज्जा करके ये बात अर्ज़ करना चाहूँगा कि लोग आमतौर पर ये समझते हैं कि असरौल से जो अजमलीन और रेज़रपीन निकला था, ये असल खोज और असल दरियाफ्त थी। मैं समझता हूँ कि ये बिल्कुल गलत है, इसलिए कि असरौल से अब तक तकरीबन 70 से 80 केमिकल कॉन्स्टिट्यूएंट्स जो है आइसोलेट किए जा चुके हैं। शायद आप अगर गूगल करें तो इससे ज़्यादा भी तादाद ये आपको बता सकता है कि इससे ज़्यादा अब तक जो है एक्टिव कॉन्स्टिट्यूएंट अब तक इससे निकाले जा चुके हैं, केमिकल कॉन्स्टिट्यूएंट निकाले जा चुके हैं। तो मैं समझ रहा हूँ कि असरौल या किसी दवा में ये अहम नहीं था कि अजमलीन उससे निकला और रेज़रपीन निकला, बल्कि उन सबसे ज़्यादा अहम ये था कि जिस चीज़ को तुम इम्पेरिकल एविडेंस की कमी कह रहे हो, तो जिन नज़रियात की बुनियाद पर, जिन शवाहिद की बुनियाद पर, जिन पैरामीटर्स की बुनियाद पर यूनानी स्कॉलर्स दवाओं की स्टडी कर रहे थे, तो असरौल की स्टडी ने न सिर्फ ये साबित किया कि असरौल जिस मकसद के लिए इस्तेमाल की जा रही थी वो मकसद सही था, बल्कि असरौल की स्टडी ने ये साबित किया कि वो हज़ारों दवाएं जिन्हें अतिब्बा इस्तेमाल कर रहे थे और जिन बुनियादों पर इस्तेमाल कर रहे थे वो बिल्कुल सही बुनियादें थी। मैं समझता हूँ कि असल चीज़ ये थी जिसको अब तक जो है तारीख़ में समझा नहीं गया, और मैं ये पॉइंट जो है ख़ास तौर पर आपको जो है ये सोचा था कि जो असल नतीजा था असरौल की स्टडी का, उस तक आपका जहन पहुंचाना चाह रहा हूँ। असल नतीजा ये था कि जो एतराज़ात थे, गोया कि वो सब के सब एतराज़ात जो है मेरे ख्याल से बेबुनियाद थे। पैरामीटर्स हमारे आपके अलग हो सकते हैं, वो अलग हो सकते हैं कि आप किसी और बुनियाद पर कर रहे हैं, मैं किसी और बुनियाद पर कर रहा हूँ, लेकिन आप सही हैं, मैं गलत हूँ, ऐसा नहीं है। बहरहाल खैर, दूसरी बात है कि उनका एक जो है दवा शादी को भी लेकर के था, मेरे साथी का इशारा है कि मैं वो कर दूं, तो मैं आपसे ख़ास तौर पर जो है दवा शादी के ताल्लुक़ से मैं आपको ये बात बता दूं कि हकीम अजमल खान ने हिंदुस्तानी दवाखाना कायम किया, और उसके ज़रिए जो उन्होंने दवाओं के स्टैंडर्डाइजेशन के लिए जो उस वक्त उन्होंने स्टैंडर्ड तय किए थे, तो मैं आपकी तवज्जो यहाँ पर मबजूल कराना चाहूँगा कि ख़ास तौर पर 1 जनवरी 2003 से मैंडेटरी कंप्लाइंस करार दिया गया है शेड्यूल टी को, जो GMP का शेड्यूल टी है आयुष ड्रग के लिए, उसके लिए जो मैंडेटरी हुआ वो ये था। लेकिन मैं समझता हूँ कि शेड्यूल 2 में जितनी भी बातें कही गई हैं वो तकरीबन वो सारी की सारी चीज़ें रेशनलाइजेशन के ताल्लुक़ से हकीम अजमल खान ने वो तमाम चीज़ें हिंदुस्तानी दवाखाना में की थी। मुझे अफसोस है कि बड़ी तेज़ी के साथ मुझे आगे बढ़ना पड़ रहा है। मैं 2 बातें अर्ज़ करना चाह रहा हूँ। हकीम अजमल खान क्या एक किस्से और कहानी की चीज़ हैं? अगर किस्से और कहानी की चीज़ हैं तो ठीक है, हमने उनको 2 मिनट याद कर लिया, उनकी याद मना लिया, बात ख़त्म हो गई। लेकिन अगर हकीम अजमल खां का जो विज़न था, हकीम अजमल खां की जो थाट्स थे, जो उनकी फ़िक्र थी, अगर वो इस वक्त भी उतना ही इम्पॉर्टेंट है जितनी कि उस ज़माने में थी, तो मैं समझता हूँ कि हकीम अजमल खां की इस सोच को, इस फ़िक्र को न सिर्फ सलाम करना चाहिए बल्कि बड़े एहतमाम के साथ हर साल यूनानी डे के मौके पर याद करना चाहिए। इसलिए कि इस वक्त जो मॉडर्न वर्ल्ड व्यू है, ख़ास तौर पर हेल्थ केयर सिस्टम का, अगर आप उसको देखें तो उसमें आपको 3 चीज़ें बहुत ही वाज़ेह सूरत में नज़र आएंगी। पहली चीज़ क्या है? इंटीग्रेटेड मेडिसिन की इस वक्त बहुत ज़्यादा बातें हो रही हैं। हर शख्स जो है खड़ा होता है तो इंटीग्रेटेड मेडिसिन को बहुत अहम उसके साथ पेश करता है। दूसरी चीज़ होलिज्म की बहुत ज़्यादा बातें हो रही हैं। तीसरी, इस वक्त बहुत हाई लेवल पर, अभी हिंदुस्तान वगैरह में तो शायद हम बहुत बाद में पहुँचते हैं, एक चीज़ जिसकी बात हो रही है वो है इंडिविजुअलाइज्ड मेडिसिन की बातें हो रही हैं। यानी ये तीनों जो चीज़ें हैं, हकीम अजमल खान ने आज के 100 साल पहले, इसलिए कि 2027 में उनका इंतकाल हुआ था, 2026 में इस वक्त हम जी रहे हैं, अगले साल उनका जो है 100 वर्ष पूरा हो रहा है, इस दुनिया से उनको रुखसत हुए। तो इंटीग्रेटेड मेडिसिन, होलिज्म और इंडिविजुअलाइज्ड मेडिसिन के लिए जो कॉन्सेप्ट, जो नज़रियात इस वक्त मॉडर्न वर्ल्ड व्यू में आपको नज़र आ रहे हैं, वो सारे के सारे कहीं न कहीं उस सोच से मुतासिर हैं जो हकीम अजमल खान ने आज के 100 साल पहले दी थी। ख़ास इसके लिए हकीम अजमल खां का नाम ना लिया जा रहा हो, मैंने उसको थोड़ा सा जो है, बहुत से हमारी बहनें भाई यहाँ उर्दू से वाकिफ़ नहीं हैं, मैंने ये लिखा है कि भले ही हकीम अजमल खां का नाम न लिया जा रहा हो होलिज्म का नाम लेते हुए, भले इंडिविजुअलाइज मेडिसिन का नाम लेते हुए हकीम अजमल खां का नाम ना लिया जा रहा हो, और इंटीग्रेटेड मेडिसिन की जब हम बातें कर रहे हैं तो उस वक्त अगर हकीम अजमल खां का भले नाम ना लिया जा रहा हो, आप बायसेंस में कुछ भी कर सकते हैं, लेकिन आपको ये बताना होगा, जैसे मैंने ये कहा था कि हकीम अजमल खां ने जो मॉडर्न क्यूरिकुलम लेकर के आए थे वो न्यू होमियोपैथिज्म की जो मोमेंट थी उससे मुतासिर था। तो ये सारे के सारे कॉन्सेप्ट अगर इस वक्त जो है डिस्कशन में हैं तो उसका रेफरेंस और सोर्स बताना होगा कि ये लोगों के जहन में बातें कैसे आईं, उनकी अपनी हैं या हकीम अजमल खां की हैं। मेरे पास इस बात के एविडेंसेस हैं, क्योंकि इस वक्त बात एविडेंसेस की हो रही है कि मैं पूरी जिम्मेदारी से बात कह रहा हूँ कि मॉडर्न वर्ल्ड व्यू इस वक्त हकीम अजमल खां की फ़िक्र से मुतासिर है। बहरहाल अब मुझको अपनी गुफ्तगू के आखिरी उस पर पहुँचना चाहिए। शायद ये बातें हमारे बहुत से ऑडियंस के लिए, बहुत सारे सामीन के लिए गैर ज़रूरी हों, लेकिन मैं ख़ास तौर पर जो मेरे यहाँ चंद साथी हैं उन तक पहुँचाने के लिए मैं ये बातें ज़रूर रखना चाहूँगा। सिर्फ 2 मिनट का वक्त लगेगा इस वक्त जो है। और यूँ भी अर्ज़ करना चाहूँगा कि चूँकि मेरी बातें रिकॉर्ड हो रही हैं, अब इसकी अहमियत नहीं होती कि ऑडियंस कितने हैं, क्योंकि हर बात जो है रिकॉर्ड हो रही है तो हमेशा के लिए महफूज होगी। तो मैं समझ रहा हूँ कि ये इख्तितामी कलिमात जो है सिर्फ 1.5 मिनट का वक्त अगर आप जलील भाई मुझे दें तो मैं शायद इस बात को अपनी जो है रख दूँ यहाँ पर। सही साहब इजाज़त है? अब हम अपनी गुफ्तगू को कंक्लूड करना चाहते हैं। अब तक आपने देखा कि हकीम अजमल खां ने वेस्टर्न मेडिसिन के उन तमाम इल्ज़ामात का जो इसके बारे में रेशनल होने के ताल्लुक़ से आयद किए जा रहे थे, इनके जवाब के सिलसिले में मसीहुल मुल्क ने बहुत ही जामे हिकमत अमली का नमूना पेश किया। ख़्वाह वो निसाब तालीम का मॉडर्नाइजेशन यानी एकेडमिक रिफार्मेशन हो, दवाओं का साइंटिफिक वैलिडेशन रहा हो, या फिर यूनानी दवासाजी को इंडस्ट्रियल ऑर्डर अता करना हो, मसीहुल मुल्क की ये तमाम कोशिशें यूनानी तिब के ढांचे को एक रेशनल पैराडाइम पर उस्तवार करने की कोशिश का हिस्सा रही हैं। लेकिन इन तमाम इस्लाहात में हकीम अजमल खां ने यूनानी तिब के कोर फंडामेंटल से कोई कॉम्प्रोमाइज़ किए बगैर सिर्फ उन्हें कॉन्टेम्पोरेरी एकेडमिक लैंग्वेज और साइंटिफिक नरेशन के साथ पेश करने की कोशिश की। उन्होंने कदीम नज़रियात का ऐसा साइंटिफिक इंटरप्रिटेशन पेश किया कि ये अपनी फ़नी खूबियों को बरकरार रखते हुए मॉडर्न मेडिकल स्टैंडर्ड से हम आहंग हो जाएं और अपनी खूबियों के साथ मॉडर्न पैराडाइम में ढलकर जदीद इल्मी दुनिया के लिए काबिल फ़हम बन जाएं। लिहाजा आज अगर हम मसीहुल मुल्क हकीम अजमल खां के कायम करदा रास्ते पर चलने का दावा कर रहे हैं तो हमारा हदफ ये होना चाहिए कि जदीद साइंसी पेशरफ्त को इस तरह असिमिलेट करें कि यूनानी तिब की स्पेसिफिक आइडेंटिटी और कोर एसेंस मुतासिर न होने पाए। हमें यह हकीकत पेश नज़र रखनी चाहिए कि साइंटिफिक वैलिडेशन की कोशिश में अगर हमने अपने एपिस्टेमोलॉजिकल आइडेंटिटी यानी मखसूस नज़रियाती ढांचे को पथे पुश डाल दिया तो फिर हमारे वजूद की मानवियत ही ख़त्म हो जाएगी। लिहाजा हमारा मिशन ये होना चाहिए कि अपनी मुकम्मल पहचान के साथ यूनानी तिब को रेशनल मेडिकल सिस्टम के तौर पर पेश करने की कोशिश करें। हमारा मकसद तालीम और रिसर्च के नाम पर यूनानी तिब को किसी दूसरे सिस्टम में जम करना नहीं, बल्कि उसे एक ऐसा साइंटिफिक इंटरप्रिटेशन अता करना है जो उसे मॉडर्न साइंटिफिक वर्ल्ड के लिए काबिल फ़हम और इंडिस्पेंसेबल चॉइस बना सके। रेशनलाइजेशन से मुताल्लिक़ मसीहुल मुल्क हकीम अजमल खां की कोशिशों का यही खुलासा है। अगर हम इस अज़्म और इस अहद के साथ इस यूनानी डे को मनाने का जज्बा पैदा कर लें तो मसीहुल मुल्क हकीम अजमल खां को यही सच्चा खराज अक़ीदत होगा। आप सभी हजरात को 10वें यूनानी डे की मुबारकबाद पेश करते हुए अपनी गुफ्तगू को ख़त्म करने की इजाज़त चाहता हूँ। शुक्रिया। अल्लाह हाफ़िज़।
Thank you, sir, for your enlightening scholarly discourse. As our guests and distinguished dignitaries for the valedictory session has been arrived, I kindly request all the esteemed speakers to please conclude your presentation within 10 minutes. Okay. Now I invite Dr. Munira ma'am, Principal and Professor, SVGM's Dr. Bhanuben Nanavati College of Pharmacy, Mumbai. She's okay. Okay, I invite you. Okay, thank you.
I'll introduce myself. So while the PPTs are on, I'll just introduce myself. I'm Dr. Munira Momin, Professor and Principal of SVKM's Dr. Bhanuben Nanavati College of Pharmacy, which is located in a Villeparle area, Villeparle East. We work on multiple Ayurvedic as well as herbal formulation. We give a general name as a herbal formulation. So I'll be just covering few case studies where we have covered how the traditional medicine, especially the Ayurvedic as well as Unani, we have used modern tools to analyze, standardize, validate. I will be going a little fast. In fact, today morning I added few slides, but I'm going to just hurriedly run through. So after a wonderful session of introduction to the, Hakim Ajmal Khan's legacy, let me wish everyone the World Unani Day. We all are know the Unani dosage forms. What we have done is studying the Unani Unani dosage form and matching up with the modern tools. So here if we see nanoparticle and nanomedicine in Ayurveda as well as Unani in AYUSH system, we have a lot of nano-formulation which we consider either as a bhashma or it is a different medications which we use in Unani also. Liposome and lipid nanoparticles, which we generally advise that the oily preparation in Unani, if can be converted into liposome or it can be converted into nanomedicine for a better penetration through skin. Controlled release, for example, morning we saw at multiple sessions, we talked about how to extend the drug release. If this particular dosage form to be taken twice a day, we can convert into controlled release by making sure that the actives are extracted, and that's how we can reduce the dosing frequency, improve the patient compliance, and that's how the novel drug delivery platforms can be utilized. So I just try to integrate the traditional and the modern technology. Is when we see the calcination, we can convert and we can further analyze it by SEM-TEM XRD, confirming the particle size and safety profile, because the particle size plays very important role in terms of elimination and excretion as well as absorption at a cellular level. As I talked about liposome and herbal active deliveries, optimized topical morning. There was a wonderful talk on a dermatological product, and I was just thinking that I will talk to the speaker and said that the dermatology, how about some of the excipient which we use, are they allowed in Unani or not? So this is in a nutshell that integration helps further in enhancing the efficacy. So case study 1, this is in collaboration with Dr. Nikhat at RRIUM Kharghar, Navi Mumbai. This is RGSTC funded project. We know that PCOS is one of the very havoc creating disorder in young girls, and majority modern Ayurveda, Unani, Siddha all are struggling to address this issue, where Dr. Nikhat suggested us the Joshanda, and we use this all combination of Unani drug, and we prepared Joshanda as per Unani method. So Joshanda in Unani method for these drugs, you soak it overnight in 500 ml water, and then morning you boil it to half, that is 250 ml, and then filter it and drink that decoction, drink that Joshanda. Now this same product I tried for my niece who is just 19 years old, and I realized that it was very difficult for her to take because of chirata. It's very, very bitter, and she had pain, stomach pain, and that's where we realized that we need to convert this into palatable formulation. So we try to convert to improve the palatability. This was our main aim. So here the objective one was to prepare decoction exactly as per the Unani method, so we don't disturb Unani principles, but the only thing what we did is we converted like this. So we prepared the decoction, the decoction was then lyophilized. So we use two methods, either a lyophilization or we use a spray drying. So spray drying, the reason was because it's a more industry scalable method, so the entire aqueous extract can be converted into powder. Or if we lyophilize, it becomes a cost intensive, the cost becomes more if we lyophilize or a freeze drying. We ultimately converted into lyophilized powder as well as spray dried because we wanted to see the actives remain same. In spray drying, we are again having a lot of velocity. We thought that there could be something changing, and that's where we saw, and we converted into the effervescent granules, and here this is how we did. We converted, or even the third method we used is the entire extract was evaporated by rotary evaporator, all the water was evaporated and then converted. Extract was adsorbed on nucilin. Now nucilin is a very simple excipient which is allowed as by US FDA, and we converted that entire thing into powder. And here you can see this is the tablet which we prepared with all those 13 hubs converted into spray dried or a lyophilized product, and that's how we worked on that. HPLC method we established because we wanted to see that if we change the aqueous quantity or if we change the boiling time, is there any change into the actives, and we saw that the actives were not affected. We established HPLC identification analytical method. Each ingredient were also identified into the aqueous content, and this is the method, and now we are going, this is the next step, we are going for a cell line study is ongoing, and we would be working on animal model. In cell line, we would be using COUNCILFOR cell line, which is very well established for PCOS model, and then we would be going for clinical trial at RRUM or the JJ hospital. I wanted to integrate nanotechnology since I thought that probably audience would be more of a Unani. So I will skip this. I'll just tell you that when there is a traditional drug, it goes across the entire body. But when it is a nanoparticle, we can target it to one particular organ. These are the various nano-formulations which can be adopted, Unani, and we sometimes think that the nano product are not scalable, industry scalable, but there are products already available in the market using nano-formulation. So another example which I give you, and I'll be skipping some of the examples now in just to respect the time. So this is one more product. As we know that India has a very wonderful biodiversity hotspots, and there are a lot of natural compounds which are used in Unani for various activities. So rheumatoid arthritis, we know that is one of the very debilitating disease. We try to prepare a nano emulsion because we know that rather than taken orally, if it is applied at the joints, it would have a better immediate, less side effects. We converted lipid Google, morning we were talking about Boswellia, and ginger oleoresin, castor oil and oleic acid. We converted with a fenugreek oil. So around 60% of oil content we could add and we could finish. So this is the last, baki all I'm going to skip. We converted into gel, oil, so this is the mechanism, and this is the gel of all those 5 oils. We did TEM image. We also did a drug diffusion study. Even the animal, we did X-ray analysis, inflammation, paw edema. We also took a histopath. We could see inflammation was reduced. Inflammatory markers were studied in comparison to diclofenac. They were reduced. And as we know, there are a lot of books are available, official drafts are available. With this, I will just stop my presentation, and basically I wanted to see that though there are a lot of competition in a Unani combination also, but if there is a modern technology being incorporated, definitely the efficacy of the formulation would be better. So I would be just skipping through, and I'll close this talk. Thank you very much. Thank you.
Thank you, ma'am, for your insightful deliberation. Now I invite Dr. Lokesh Kumar Bhatt, Professor, Department of Pharmacology, SVMs, Dr. Bhanuben Nanavati College of Pharmacy, Mumbai. Sir, please conclude in 5 minutes. Okay, sir. Thank you, sir. क्या चलेगा?
Good afternoon. So my job is to finish within 5 minutes. So I will try, sir. So I'm a pharmacologist, so maybe I'm kind of slightly out of place, but not out of the scope completely. I will just start with one quote by the Mr. Abdul Majid, who is chairman of the Hamdard, and they are the forefront in the all those Unani medicines. So what he said recently, that Unani medicine today stands at unique crossroads where tradition and modern science can enrich one another. So this is one of the lifelong thing which we can do, where we can actually collaborate with each other, and there can be synergy in the outcome. So quickly, that what are the evidence-based validation include? So there are clinical research and trials, then validation of fundamental concept, pharmacological evidences, and what are the challenges here? So there are main challenges which actually everyone is right. Unani Medical, they are right, and as a pharmacologist, we are also right. For example, one of the gold standard in the pharmacology for testing is the randomized clinical trial. But the challenge, for example, is the placebo challenge. Now we consider placebo is one of the must, but that can be a challenge with the Unani. For example, you can take case of the Jaswanda. Now Jaswanda contain all the smells, all the fragrance. You cannot create placebo like that. And that is a genuine problem where it is very difficult to create such kind of trials. So there is need for some kind of specialized trials which need to be created to overcome these kind of genuine differences. Same way there are, I will quickly bypass, there are some other kind of validation which should be done. So whether the reverse pharmacology is the key, in a way, yes, because in this case we know what is the drug is being used for, so that makes job less easy compared to our drug discovery pharmacology, where we start from the scratch. So reverse pharmacology is good way to save time and money. So Unani medicine, it can be actually in the pharmacological way, can be started with the in silico studies, where we can go with the network pharmacology, followed by the in vitro studies and the in vivo studies. So quickly, again, network pharmacology is one of the recent phenomena. Chinese, they have expertise, they have come out with some of the very good phenomena and some of the very good drug out of that. So network pharmacology will be like this. So we will have drug, we know in case of, the challenge here is in case of Unani medicine, we have many drugs together. So we need to make a big list. So again, there is library available. Government of India made that library. If not, we need to come out with those ingredients and then compare it with the target of whatever disease is there. And then there are number of those softwares we need to use where it gives the protein-protein interaction and most probable targets which can be targeted by that particular kind of formulation containing so many of the compounds. So with that we can get the most probable targets for the particular disease, and then with those best targets we can actually dock the molecule and we can come out with the best score and best targeted approach which we should do. And then same way then there can be molecular docking and the MD simulation. And then additionally there can be ADMET, which is actually not very much required in case of the Unani formulation because they are well tested, but we can actually do the ADMET and we can produce the data to actually corroborate with the earlier one. And then it can be in vitro, where mainly means all those studies, apoptosis, cell cycle, all those function-based assay can be done. And then comes the main in vivo studies, where mainly the in vitro, the toxicities, using all those guidelines, and in vivo efficacy studies can be done, where the main change is that rather than simply doing the efficacy, we can go in the detail studies where we know that what is happening in the body. For example, we can go for the detailed biomarkers, gene mapping and all those studies, so that we know that after taking that particular medicine containing so many of the compounds, what is happening. And then pharmacokinetic studies and clinical trial, what is the main change which should be done there in the Unani medicine is the mechanistic, which can be in the less number of patients, but mechanism should be known. That is the main challenge, and that is what we can do. That's it, sir. So there are pillars. I will just quickly bypass it. Yeah. So I will just at the end I will share this study, which we did the Eucalyptol, where we just went in this direction. So initially that network pharmacology, followed by the docking, and then the hamster, the atherosclerosis in the hamsters, and then we came out with the mechanism-based action of the Eucalyptol for the action against the. So that is, I made it very crisp and quick. I want to acknowledge all those agencies like the BRNS, DST FIST, DST WISE, AICT, SVK Management, and our team DNCP, and our principal for all collaboration and supports. Thank you very much.
Thank you, sir, for your pharmacological insight. Now I invite Dr. Bilal Ahmed, sir, Research Officer, Unani, Scientist Level 4, Hakil Haram, New Delhi. Sir, please.
कहाँ वो लगा ना पीपीटी लगा ना अरे पीपीटी तो लगाओ भाई 5 मिनट पहले बता रहे हो बात शुरू नहीं हुई बंद करो मुशायरे का आखिरी शायर भाई तुम हाँ सही ये बात आपने सही कही Respected chairperson, distinguished guests, delegates from all across the country, colleagues, it is both an honor and a defining moment to show how the literary research program of the Council at Hakim Ajmal Khan Institute for Literary and Historical Research in Unani Medicine is bridging the classical legacy with the demand of today's health scenario. So we will discuss only 3 programs, that is National Unani Morbidity course, and then the inclusion of Unani medicine and Ayurveda and Siddha in ICD-11, and then the new project, that is International Classification of Health Intervention. So you are well aware that literary research program is the one of the programs of the council, and which is related, it supports education, research and evidence-based practice, contributing to the integration of Unani medicine into existing healthcare. So the initiatives by Ministry of AYUSH, that is joint initiative of the Ministry of AYUSH and the World Health Organization, have positioned the CCRM and its peripheral institute, Hakim Ajmal Khan Institute for Literary and Historical Research in Unani Medicine, as a key contributor to national and global standardization in Unani Medicine. The institute played a central role in developing national Unani morbidity codes. These are the codes which have been assigned to the morbid condition discussed in the classical of the Unani medicine, and their definition has been standardized. So this national morbidity codes were implemented through Namaste portal, that is the National AYUSH Morbidity and Standardized Terminologies electronic portal, that is the reporting portal, and the second one is the AYUSH Hospital Management Information System. So first morbidity was classified, then again it was given a code and its definition was standardized, and then this code was used for reporting the morbidities. This effort of the Ministry of AYUSH for implementing of National Unani Morbidity codes led to the inclusion of our morbidity codes to the ICD-11, as WHO wants the thing that you are running your national program for 2 years. So we have run our national program, National Unani Morbidity code for 2 years, and then the analysis was made and that was included in the ICD-11. The development of National Unani codes during 2017 represents a major step towards integrating Unani medicine with digital health systems and bridges tradition with technology, enhancing the credibility, visibility and future growth of Unani medicine. That is ICD. You are well aware that ICD is a record and report health and health related conditions which reports globally. So traditional medicine chapter 1 is related with the Chinese medicine. I am running through all these things because time is running out. So ICD 11 has its own structure, but in the 11th revision they have included the traditional medicine. So traditional medicine module 1 is related with the Chinese medicine with the collaboration of China, Japan and Korea, and the traditional module 2 have been assigned to Ayurveda, Siddha and Unani. So we have done, Minister of AYUSH has done a MOU with the WHO, and we have worked for 4 years, 5 years I think. I have been the part of the team, and Dr. Neelam Qudusi, who is not here, Dr. Aman Sahab is not there, he is also a part, Dr. Ahmad Saeed, Dr. Mirajul Haq, they have been part of that. So this is the, I am unable to see here, but this is the journey from 2020 to 2025. Now in 2025, no, it's okay. In 2025 February, the official release is in WHO Blue Browser. Blue Browser means that it is ready for reporting now. They had two types of browser, one orange, that is for foundation and maintenance, and the blue browser is for reporting. So now India is very much ready for implementation of this TM2 module with the mapping of these codes of traditional module 2 with our own national immunology codes and the biomedicine codes of the ICD-11. So these three go hand in hand. This is something, these are the use cases and benefits. Enhanced patient-centered care, promotion of evidence-based TM, facilitating cross-cultural collaboration, public health integration, systemic recording of TM morbidity codes. It is implementation. There are difficulties, workforce is needed, capacity, challenges, and future roadmap. Now a new project is coming from WHO with the collaboration of Ministry of AYUSH. That is International Classification of Health Intervention. It is a World Health Organization's framework designed to standardize the reporting analysis of health interventions such as diagnostic, treatments, and rehabilitation. ICHI, it covers a wide range of services including primary care, acute care, and traditional medicine, serving a tool for health statistics, research, and comparative analysis. Its purpose is to provide a common international language for tracking and evaluating health intervention, improving patient safety, and strengthening the health system. ICHI is based on the three structure is three axes. One is target, that is anatomical structure. What are you doing at which organ? And which organ you are doing? Then action, what are you doing? And means how are you doing? These three axes made ICHI. So as we know that leech therapy is very much prevalent in Unani medicine. When we discuss leech therapy, site unspecified LZZ DBHAH. So this is defined as external application of substance or living organism to skin and subcutaneous tissue. This is the major classification, and leech therapy is the child of that one. So target is here is LZZ, integumentary system target. Action is DB, application of substance, and means is AH. So when we say leech therapy, it is the code is LZZ DBAH. Same is cupping here. Cupping is PZX SRAH. PZX is here, any site not elsewhere classified. Action is SR, cupping, which is defined as suction by using a vacuumized cup or jar. AH is external. So there are 2 or 3 codes only for traditional medicine now, but we are going to develop a national codes for our intervention here in India. We will go for it for 2 or 3 years, then we will convince the WHO to include our codes with their own ICHI system. The release of the TM2 module in the global diagnostic browser, an initiative to include a ASU system into international classification of health intervention, is more than a technical update. It is a landmark achievement that elevates indigenous knowledge to a formal global standard. This synergy between national digital infrastructure and international health frameworks ensures that traditional systems are not just preserved as heritage, but are actively integrated into the future of global inclusive healthcare and insurance system. This is the glimpse of the blue browser. You can see here the traditional module inside. That is module 1 and module 2 is there. The next one is when we search humma, that is fever in Unani medicine, these list comes and you can report accordingly. So thank you very much for your patience hearing. Thank you. बहुत brief रहा मैं
Thank you, sir, for your insightful deliberation. Now, I request our respected chairperson to kindly moderate and share their valuable observation in short.
Hello. First of all, thanks to organizing committee and all authorities of the CCRM for giving me such type of opportunity to chair the session. And it is most difficult to conduct afternoon session to participants and chair also and speakers also. Due to short of time, I am expressing my views very short and very in the conclude manner. This session has clearly demonstrated how Unani medicine is progressing towards a stronger evidence-based framework while preserving its classical foundations. First lecture of the presentation by the Dr. Amanullah from Amanullah Research Officer from Central Council for Research in Unani Medicine, New Delhi, highlighted the systematic journey towards developing standardized Mizaj assessment tools. CCRM, along with guideline and references work supported by the World Health Organization, has significantly contributed to drug standardization and quality assurance in Unani medicine. Second lecture delivered by Dr. Fakhri Alam sir on Hakim Ajmal Khan reminded us of his great contribution as a physician, educationist, freedom fighter and visionary farmer who worked tirelessly for the traditional development of Unani system of medicine continuously. And the third one lecture is the presentation on advanced pharmaceutical pharmacological technologies, emphasize the need for innovations, quality assurance and modernization of Unani therapeutics. As coordinator of elective course under the National Commission of Indian System of Medicine, I strongly believe that continuous innovation and research development are essential for the future advancement of our system. Fourth lecture delivered by Professor Lokesh Kumar Bhatt on pharmacological exploration of Unani medicine through biopsy and in experiment was highly scientific and forwarding looking, integrating laboratory based biopsy with molecular level validation provides a strong scientific evidence and bridge traditional knowledge with modern pharmacological system. And the last lecture के बारे में मैं बहुत मतलब मीन्स नहीं बोल पाऊँगा क्यों, तो मेरे वो टीचर है, मैं टीचर को क्या एनालिसिस करूँ, लेकिन जो उन्होंने अच्छी चीज़ें बताई, वैसे अमानुल्लाह सर से भी मैंने बहुत हेल्प लिया था अपने स्टडी के ड्यूरेशन में, तो मैं बहुत डर के मतलब इसलिए बोल रहा हूँ कि वे सब मेरे बड़े लोग हैं और बहुत सीनियर हैं, लेकिन जो व्यूज़ थे जस्ट लाइक फॉर द सिस्टम मैं एक्सप्लेन कर रहा हूँ. बहुत अच्छा लेक्चर था उनका. The fifth lecture by the Dr. Bilal Ahmed, Research Officer Scientist at Central Council for Research in Unani Medicine on evidence-based initiatives under the literary research program, highlighted the importance of systematic documentation, critical analysis of classical text, and research-oriented interpretation of strengthening the academic foundation of Unani system of medicine. In the last, as a coordinator, as a chairman of the electives course on the advanced innovation in the Unani system of medicine under the aegis and under the collaboration with National Commission of Unani System of Medicine, I firmly believe that innovation, scientific validation and research integration are essential for the future growth of our system. I sincerely congratulate all the speakers for their excellent and meaningful scientific presentations. Thank you to all.
Now I request to my session chair Anwar Saeed sir, Ahmed Saeed sir, for deliver some key points for this session.
थैंक्स डॉक्टर जलील साहब। मैं आप तमाम स्पीकर का सबसे पहले शुक्रिया अदा करना चाहता हूँ, और अपने ज़्यादातर जो लेक्चर हैं वो मेरे साथी और मेरे अहबाब ही हैं, और अहबाब और साथियों के बारे में, कलीग्स के बारे में, पीयर्स के बारे में कुछ कहना यकीनन बहुत ही मुश्किल काम है। फिर भी मैं अपनी ज़िम्मेदारी को निभाते हुए मैं अपनी बात को बहुत ही मुख्तसर तरीन तरीके से ख़त्म करूँगा। पहला लेक्चर हमारे कलीग साथी डॉक्टर मानुल्लाह साहब का था। डॉक्टर मानुल्लाह साहब इल्मी हल्के में और यूनानी दुनिया का एक ही बहुत ही मोतबर नाम है, जिन आपने मिजाज के ऊपर, मिजाज के लफ्ज़ी साख्त से लेके उसके डिजिटल सफ़र तक को नहायत ही खूबसूरती के साथ पेश किया। सेंट्रल CCRUM के लिटरेरी रिसर्च इंस्टीट्यूट के आप एक तरीके से रूह रवा हैं, और उसके प्रोग्रामों की न सिर्फ मनसूबाबंदी करते हैं बल्कि वहाँ कोई कामों के बाज़ इशात को भी आप ही देखते हैं। हैरत होती है कि आप इतनी हुकूमती ज़िम्मेदारियों और हेडक्वार्टर की मशगूलियात के साथ साथ इतने उम्दा और ख़ूबसूरत पेपर को न सिर्फ पेश किया बल्कि बहुत ही दिलनशीन और दिलावेज़ अंदाज में पेश किया। मैं इसके लिए आपको मुबारकबाद देता हूँ। उसके साथ दूसरा लेक्चर मेरे साथी हकीम फ़ख्र आलम साहब का था।
Sorry sir, but I request to brief
बस
you.
1 मिनट।
Okay sir, thank you.
और 2 मिनट। फ़ख्र आलम साहब इस नौजवान नस्ल के सबसे ज़्यादा जूद नवेस और कसीर तसानीफ मुसन्निफ़ हैं। आप कम से कम 5 से 1 दर्जन किताबों के मुसन्निफ़ हैं। आपकी खुसूसियत है कि आप हर मौजू पे नहायत ही नया नुक्ता लेके आते हैं, और अभी भी आपने इसमें जो पेपर पेश किया वो एक नुक्ता रस और नुक्ता अफीनी आपके मिजाज में है। यूनानी हिस्टोरियोग्राफी या तारीख़ नगारी की तनकीदी तारीख़, क्रिटिकल अप्रेजल आप बहुत अच्छी तरीके से, बल्कि ये कहा जाए कि इस नस्ल में बानी हैं, अव्वल हैं, तो बेजा नहीं होगा। डॉक्टर बिलाल साहब मेरे वो साथी हैं जिनके साथ मेरा वक्त घर से ज़्यादा उन्हीं के साथ गुज़रता है। तिब्बे यूनानी को बैनुल-अक़वामी लेवल पे ग्लोबलाइजेशन में ले जाने में, WHO के साथ काम करने में, और अभी सेक्रेटरी सर ने जो अपने कारनामे को गिनाया उसमें ICD 11 का तस्करा था। उसके पीछे अगर ये कहा जाए तो डॉक्टर बिलाल ही रूह रवा हैं, और वही उस काम के और उनके दो बहुत मुख्तसर टीम के साथ ये काम करते हैं। प्रोफेसर मुनीरा मोमिन फार्मास्यूटिकल दुनिया की एक मोतबर नाम है, और आप नानावती कॉलेज में हैं। आपके फार्मास्यूटिकल पे तकरीबन दर्जन भर पेपर हैं। दवाओं के इस्तेमाल को सफूफ से लेकर नैनो पार्टिकल का किस तरीके से इस्तेमाल हुआ, आज उसको नैनो पार्टिकल की शक्ल दे रही हैं, और काउंसिल से कोलैबोरेशन में उसके बहुत सारे प्रोडक्ट्स को आप एक नई जहत, नई डायमेंशन अता कर रहे हैं। आपके साथ साथ डॉक्टर लोकेश कुमार साहब, आपकी सबसे बड़ी खूबी है कि अपने पूरे काम को और इतने अहम काम को सिर्फ 5 मिनट में समेट दिया। ये भी बहुत बड़ी खूबी है। इन सब के साथ मैं तमाम लर्नेड ऑडियंस को मुबारकबाद भी पेश करता हूँ, और इस सेशन को खूबसूरत तरीके से कंडक्ट करने के लिए डॉक्टर यास्मीन को भी मैं मुबारकबाद पेश करता हूँ। डॉक्टर फरहत जो काउंसिल में अभी नई हैं, उनकी भी को भी मैं मुबारकबाद पेश करता हूँ कि बहुत ही खूबसूरत अंदाज में सेशन को कन्वे किया। थैंक यू।
Thank you sir. Now it's the time for facilitation. Okay. I will. Okay. Okay, sir. I request Dr. Yunus Iftikhar Munshi sir to facilitate Dr. Jalil Ahmed sir with flower and memento. Okay. I will request Dr. Munshi sir to facilitate Dr. Ahmed Saeed sir with flower and memento. Thank you, sir. Thank you. Okay. Okay. As a token of appreciation, I would like to invite Dr. Amanullah sir, and I will request Dr. Jalil sir to present memento. Thank you, sir. Now I invite Dr. Fakhri Alam sir to receive the memento. Okay. Now I request Dr. Munira Momin ma'am to kindly come on stage and please receive your memento. Thank you. Now I would like to invite Professor Lokesh Kumar sir to please come on stage and receive your memento. Okay. Now I invite Dr. Nigha Tanjum ma'am to please come on stage and present the memento to Bilal Ahmad sir. Okay. Okay, sir. Thank you, sir, for be seated now. On behalf of the organizing committee, I sincerely thank our respected chairpersons, esteemed speakers, and active participants for making the session intellectually enriching. Thank you all. Before leaving the room, please, I have a group photograph. Yes. Okay. And after photograph, I request all the esteemed members to reach Hall A for valedictory session. All the speakers, please come on stage. And rapporteurs, Ashok sir, please come on stage.